were absolutely, I don't know what is going on here, they're absolutely upregulated. So also this is further stressed by the fact that after silencing, also there is a significant higher protein expression of the metalloproteinase 2 and 9, which they are involved as well in the extracellular matrix degradation and in the EMT and in the transition, epithelial-mesenchymal transition. So everything hinting at the fact that when you downregulate PI-PLC beta 1, sorry, doesn't move. Why? Beta 1, the cells, they do acquire a, and also both in both cell lines, we found that using, using the Transwell migration assay as well as the Wood-Elling migration assay, we found that PI-PLC beta 1, when silenced cell exhibited an increased migration compared to the control and the wild type cells. And also for the wound healing is the same. The silencing is responsible for a very increased repair of wound after 24 or after 48 hours. And again, looking at invasion test, we also saw that PI-PLC beta 1 silencing cells, they did exhibit an increased invasion compared to control and wild type cells. It's immunocytochemistry and percentage of invading cells, you know, both in U87 as well as in U251 cells, a dramatic increase. And another test, which is the migration and gel tracts invasion assay, we did that on primary human astrocytes, so not in those two types of cell lines, but in primary human astrocytes. And also in this case, we found that the silencing of PI-PLC beta 1 was absolutely important, was inducing a great increase in the migration and the invasion compared to the wild type. And the same is for wound healing migration in those primary human cells, astrocytes. Again, when we knock down PI-PLC beta 1, PI-PLC, the migration is more and more active. And also looking at some markers of progression of tumorigenicity like Ki67, we did this evaluation by immunofluorescence, we found that actually there is both in cell lines as well as in primary cells, we found that there is an increased expression of this marker when PI-PLC beta 1 is downregulated. And also looking at beta-catenin, again, just to cut a very long story short, is important issues that we have a nuclear presence of active beta-catenin in PI-PLC beta 1 cells, respect to what happens in control cells. So just to have a take-home message, we can say at the moment that all the clinical data collected on glioblastoma, fresh and frozen samples, together with cellular and molecular data that we have done, engineering the cells and making tricks on the cells, knocking down PI-PLC beta 1, they do suggest that the downregulation of this enzyme is in some way important for physiopathologic alteration, inducing what? Inducing the cells to acquire a greater capacity of migration, invasion, proliferation, and survival. And actually, this is something which parallels very well what happens in patients in which we do see that, in glioblastoma patients, which we do see that the PLC beta 1 is downregulated. And just I want to finish thanking the lads and lasses in my lab. I beg your pardon for very very English way to nominate them, and the collaboration that we did have in the past and also in the present. And right now, I want to underline for this mainly the collaboration with Pangil Su, and that's the one with Jim McCovery. I think they are listening to us, and I thank you for attention, and I'm here to take questions. Thank you very much.
So thank you, Professor Cocco. The floor is open for questions. I think we'll take questions in the audience first and then off the screen, please. I'll repeat the question. I'll repeat the question.
Hello. Hi, Lucio, you gave an excellent talk on PLC beta, that it's reduced in the glioblastoma or glioma. But did you see which type of glioma is that? Means that the secondary or primary, or did you see any other genetic abnormalities which you found? Because where you show all the individual tumor, I could see the range. They were a lot down, but they were up also, some of the samples. Was this correlated with anything like a the new classification of glioblastoma, neural, proneural, mesenchymal, or classic type? Any of these associations did you find, or anything like a P10 abnormality or loss of chromosome 10 or the EGFR amplification?
Yes, thank you for the question. Yes, with the guys of pathology department, we have been checking all those things, and actually quite a lot of them, they do have a P10 mutation. They have a number of chromosomal abnormalities. So the fact is that, as you know, all those abnormalities cannot be linked directly to the progression of the gliomas. Yes. And so that's why we focus on this, which seems to be common to all different types. So that all different types of glioma, even though we are carrying out more and more deeper studies along with the patients, because, you know, the majority of patients are coming from are those which we do have the frozen samples. So we are checking all those things, absolutely.
Ohad.
So I actually have two questions. The first, I know that PI-PLC-P1 is typically associated and studied in neuroglioma, as you showed us, but I wonder if it's also expressed in other cell types.
Oh yeah, it's more or less ubiquitous. It's expressed more or less in all cells, in every type of cells. But in the nervous system is one in which all the PI-PLCs, gamma 1, beta 1, they are highly expressed in mainly in the nervous system.
Okay, and I wonder if you looked on the expression profile during cell cycle.
Okay, we looked at the expression during a cell cycle in other cell types, not in this type of
cancer cell you can look on.
Yeah, and actually no, we never found a real, a trustable difference in one moment of the cell cycle. So it's mainly a question of activity. And Julia did a wonderful job on, for example, in myogenic differentiation. And she made a mutant with localization, nuclear, etc., everything fine, but losing the phospholipase activity. And then that mutant blocks myogenic differentiation. So what she did in that type of assay.
Great presentation, Lucio. If I understood correctly, some other isoforms of PLC were also downregulated. Did you check if the production of PI4, P2 was decreased?
Okay, that's the problem is to check the real amount of PIP2 is really very, very complicated. You know, we did that in the past. Is a friend of ours, Naldevich, did this measuring. It's complicated, and the reality is that some other phospholipases are also downregulated as well, and many the delta, one of the delta family. Gamma was only downregulated in frozen samples, not in fresh samples. But yes, we are furthering the analysis on the other phospholipases of delta. They're very, Stefano, correct me if I'm wrong, but we have very, very preliminary data showing that the same trick that we can induce with the knockdown of PIPLC beta 1, we cannot induce knocking down the other phospholipase. But okay, it's very, very preliminary, absolutely.
Okay. But maybe the progress in mass spectrometry now maybe will allow sometime soon.
Yes, it will be very, very helpful.
Well, I think the problem would actually be getting the nuclear pool, right? That's the problem, right? To measure the nuclear pool of PIP2, and then mass spectrometry almost serves as a disadvantage there, because mass spec sees everything. Contamination's the problem. No, I get that. The question is how effective is the separation? I mean, trace contaminants.
Yes, yes, that's the problem, because you know, when you purify nuclei, you can lose a lot of things. So we did something actually all together, and because if nuclei are not really highly purified, okay, it's a cytoplasmic contamination, but if it is highly purified, you lose a lot of it, and we don't know exactly what we lose. Am I correct? Yeah.
That's right. I think the question's a great question. The technical problem is a big one.
Indeed.
So actually, you were very much on time, so we have time for extended questions. I have one. So the genetic profile of glioblastoma has been published, and actually the driver mutations appear to be histone 3, histone 3-1.1, 3 K27M, I think. And then also I think there's a BMP pathway activating mutation. So I'm curious, if one goes back to those data, does one see PLC beta come up as a glioblastoma risk gene? You would expect that would be the case, because loss of function in one allele should probably increase risk. So I'm curious if that's just been looked at.
Yes, that's a very, very, very curiosity for me too. Also in this case, it's not that easy. You know, one other thing I always asking them to try to do, but it looks to be quite complicated, is just to do exactly what they did in DM1 and DM2, to knock in PIPLC beta 1 in primary cells. They tried, isn't it? But quite complicated. That would be a good way to say, okay, I can restore, if that is true.
Are there questions from the online audience? I can't see that. If there aren't, I guess, Mina, we can go, but we should give them a chance.
Mina?
Is there anything on the screen?
Sorry?
For chat, on questions? I don't know.
It should be yes. Chat here. Okay, this one from Aurelia Lewis. Have you looked at changes in splicing when PLC beta 1 is silenced? Not yet. That's the answer. Very quick.
Which chromosome it is localized to?
Which
chromosome is the location? Location
20 is chromosome 20, the short end of chromosome 20 in the region 2021p.13. That's the region. Short arm of chromosome 20. That's the location of this phospholipase.
Okay, I think if there are no further questions, good discussion. We'll thank Professor Cocco again. And
thank you.
And we'll move on to the next speaker. We will stay on the same topic of phospholipases. Dr. Pangil Su from the Korea Brain Institute in Ulsan is our next speaker. And at about 9:30 p.m. local time, his local time, he will speak to us about genetic alterations of phosphoinositide-specific phospholipase Cs in brain disorders. Pangil, welcome.
Yes, thank you. I will try sharing. It's okay?
It's good.
It's good? More?
One moment, Pangil.
It's okay? Hear me? Chairman? Can you see my slide?
We-
Please.
Pangil, can you share your screen? We can see you.
You can you see?
Good.
Yes,
Hear me?
yes, we can hear you.
Okay, I will start. It's okay?
Yes,
Okay. Yes, thank you, Chairman. Hi, hello. So I want to say, although it is pity that we can't meet all together at the Bologna in person due to Corona pandemics, I'm very happy to attend the 62nd ABR meetings and would like to thank Dr. Lucio and his lab members for preparing this meeting indeed. Today, I'm going to talk about prediction of genetic alteration of phospholipase isozymes in brain disorder. In my talk, firstly, I will introduce genetic variation in brain, and then GWAS deep learning and prediction of cryptic variation of PLC genes. Finally, I will suggest the possibility and expectation for GWAS-A1 fusion research in perspective. This slide shows that single nucleotide variants, SMV-derived alternative splicing generated protein diversity in the cells. As you know, that elucidation of genetic variation in various human disease is one of major scientific challenges in 21st century. In this picture, normal process, but sometimes messenger RNA occurred alternative RNA splicing. This event finally makes abnormal proteins. Therefore, abnormal splicing is bring about unusual cellular functions. Up to now, 16 PLC isozyme has been identified in mammals. So based on sequence similarity, they have been divided into 7 types: beta, gamma, delta, epsilon, zeta, and atypical PLC XD, likely considering ligand-mediated response, calcium dependence, and the localizations. They are also classified into the primary PLC, immediately responsible to stimulation, and the secondary PLC for amplification of second messenger. In all mammalian PLC, highly conserved region are catalytic XY domains, as well as various regulatory domains, including C2 domains, EFN, motive and Braxton homology domains. Firstly, in 1998, mutations in human PLC delta 1 genes was reported from DNA sequence analysis in Alzheimer's patients. It is shown that 80 patients occurred mutations. One single mutated nucleotide G changed to A at 105 arginine. It means, in essence, that this arginine coded CGC changed to histidine coded CAC at pH domain for PLC delta 1 gene body. In these two papers, large-scale genome-wide analysis has identified risk genes for bipolar disorder, namely, as one of candidate gene is PLC gamma 1. As you can consider to resolve these issues, knockout mice will be provided as good model to understand pathophysiology of bipolar disorder. In my lab, from PLC gamma knockout mice, we observed that imbalance of excitation and inhibitions leads to abnormal manic behavior, such as bipolar disorder. These results suggest that dysfunction or mutation of PLC gamma 1 might cause neuropsychiatric disorders. In 2014, UK groups reports clinical research that PLC beta 1 mutations was involved in the severe early infantile epileptic encephalopathies of 10 months female infants who broke out multiple times seizure every day. From microarray data set analysis of bladder, and then found single nucleotide polymorphisms at chromosome 20 of PLC beta 1 genes, exon 1, 2, and 3. In comparison of single nucleotide 90 in father and infants. Father has adenine or guanines, while infant consistently adenine A. So, this data indicating this instance occurred in developmental stage. From analysis of human genome database, this slide shows a comprehensive summary of PLC gene alterations to be involved in brain disorder such as Alzheimer, epilepsy, autism, and bipolar disorder, and so on. As you know well, in these days, genome-wide association study, GWAS, is widely used for identification of pathogenic and genetic variations in several diseases. This technique can find small variation in genome, so-called SMV, which occurred more frequently in people with a particular disease than in people without disease. Additionally, may evaluate the person's risk of developing a certain disease. This is the reason why GWAS is popularly one of way for scientists to identify the gene involved in human disease. Recently, Netherlands groups identified 24 genomic loci linked to variations for Alzheimer's, ADHD, and schizophrenia from big data analysis of GWAS. As you know, GWAS can estimate chromatin accessibility and histone modification, DNA maturation, evolutionary conservation patterns, transcription factor binding, specificity, as well. Therefore, GWAS comes to be useful approach to investigate the relationships between disease and genetic information. In 2016, this research groups found de novo mutation in human autism through the GWAS-based studies. They shows that cluster de novo mutation in autism spectrum disorder (ASD). This data suggests the ASD mostly originated from its mother, as considering gene9 mutations. Through gene9 mutations, eventually GWAS can validate de novo mutated regions and nucleotide alterations related to genomic genetic disease. In another interesting report from Science in 2018, they found that approximately 2,500 somatic SNV in mature single human neurons. It means, in essence, that somatic SMV increased with age in the prefrontal cortex and dendrite gyrus. Taken together, unique GWAS-based techniques has gradually developed in the power with the meta-analysis, or AI identifies unknown genetic events in human disease. This table summarized PLC isozyme mutations from human genome mutation database, providing good information for gene regions responsible for human inherited diseases. A mutation in PLC isozymes are highly related to various human disease. As you want it, you can freely access database through the HGMD website. As shown in this slide, GWAS has pros and cons. GWAS provides lots of benefits, such as identification of novel SMV, discovery of novel biological mechanism, diverse clinical applications, easy to share data, and so on. However, have several limitations because of technical restrictions. Now, to overcome the limitation of GWAS, scientists apply deep learning computational techniques. In these days, artificial intelligence, AI, is already embedded in our daily life. As case in point, Google DeepMind's AlphaGo programs defeated the South Korean Master Lee in board games Badu. Essentially, deep learning is derived from machine learning method based on artificial neural networks. Now, combination of GWAS and deep learning has accelerates the discovery and the prediction of several new genetic variation in disease. Sequence-based deep learnings help to assess the significance of such variants and provide prediction of genotype and phenotype correlations. Particularly, modeling transcription factor binding sites prediction using convolutional neural network can give important information to understand how to gene regulations. In sequence, I will explain one of deep learning based prediction algorithm, so-called supervised learning. Normally, Supervised learning use training sets to teach both model to yield desired outputs. So first of all, so let me briefly introduce supervised learning. So we put dark data set. At that time, this program learns various features of dark. And next, we input non-dark data sets such as rabbit and hedgehog and so on. And then program learned feature of non-dark information. This algorithm for predicting dogs is completed through the thousands of repetitive learning. So finally, when you input different species of dog features, this algorithm can predict dogs accurately. One outstanding article has come out in 2019. There is developed how to predict cryptic variations named the Splice A program, A1 programs in first times. Splice A1 utilized deep learning based tool and the supervised learning to identify cryptic variations. Cryptic splice variants eventually disrupt normal pattern of messenger RNA splicing. Basically, splice A1 predicts genomic sequence that score various genetic position in pre-messenger RNA transcripts. Here we identified PH-Cameron cryptic variants using the learning-based Splice A1 systems in 2021 report in PNS. This algorithm takes free messenger RNA sequence as input and return splice acceptor and donor score between 0 and 1 for each position in sequence. So 1 means 100%. And we found 80 specific cryptic variation of PLC-gamma1 involved in abnormal messenger RNA splicing. This data from mouse genome was confirmed in human AD samples using Splice AD database using Splice A1 in the first part. First of all, we performed high throughput total RNA seq from Alzheimer's model mouse brains, and then screen PH-gamma gene expression patterns. PH-gamma gene expression levels slightly increase Alzheimer's brains, but not significant change. Therefore, we try to find SMV mutation from gene expressions, and then identify some putative mutations. We found 14 Alzheimer dependent SMV at PH-gamma1 gene body. Interestingly, one SMV at exon 27 accompanying frame shifts and dense mutations occurred in evolutionary conserved regions through all species. So meaning that this nucleotide is important for maintenance of homeostasis. Subsequently, we did the Splice A analysis through human pre-RNA transcripts, and then we analyzed statistically comparison of splicing events of PH-gamma1 genes. Very interesting. Some SMV is likely to be related to abnormal splicing in Alzheimer's mouse brains. This study can accurate prediction of splicing in given reference the genomic sequence. From this study, analysis of big data of mouse and human with the Splice N A1 algorithm shows accurate performance for prediction of splicing alteration to affect very affects various brain disorders. Conclusively, we have established the first concept of deep learning models to identify Alzheimer-specific SMV from combinations of genomic analysis and A1 tools. By using splice A1, we identified new splicing disruption score and alteration positions for PLC isozymes. We will expect that this data will give good information for understanding PLC isozyme related to various human disease. In the last one, I'd like to suggest possibility and expectation GWAS AY fusion research. Significant progress in high throughput genomic analysis has been made in predictive biomarker of how SMV and translate protein affect disease. However, limited supply of human materials such as brain tissue from patients, and then their questions remains unanswered. Suggestively, deep learning-based splice A may offer unique opportunity to development of novel potential targets brain disorder. So to be sure, GWAS A1 fusion will become useful tool for genetic analysis and clinical applications. Okay, I want to say I'd like to acknowledge those who participate in this presented work. I really appreciate Dr. Chu, JL, KBRI, and his lab members, my friend Lucio, and other colleagues. I will stop here today. I hope that my presentations provide some insight helpful to your research. It will be my great pleasure to meet you in face-to-face manner in soon. So hopefully you and your families are doing safe and fine during this corona pandemic. Thank you very much for your attention. Okay, thank you.
Thank you very much, Pangil. So the way I suggest we proceed is that you look on chat to see if there's any questions there. And then if there isn't, we'll take questions from the audience here. Okay?
Okay. Okay, Dr. Cha.
Anything on chat?
No. I will try.
Oh, chat is empty. Questions from the audience.
Okay, yeah, I'm sorry.
Yes.
Yes.
Great talk.
Yes.
Among all the mutations you found, I think most of them are probably loss of function mutation, but could you predict if you have some gain of function of any of those mutations?
Oh, yes, not yet. So I expect some mutation, so provide gain of function. I expect. So now we identified most of loss of functions due to mutations. I expect some mutation get the gain of function.
So Pangil, this is Vitas. I have a question. So when you train a deep learning algorithm, do you actually keep some of the data aside and not put it into the training program and then go back and ask how well the program can pick up these other mutations? I guess what I'm wondering is, how does one prevent this becoming a circular argument that induces bias very early in the learning program?
Okay, I'm sorry. I am not expert. This learning program develops. So develop the algorithm. My colleague, young researcher in my institute, Dr. Zhu. His expertise. So he basically he major in information and statistical. So I'm sorry, I have no idea. So I ask my colleague, so your questions.
Thank you.
Okay.
Anyone else?
Mhm.
There might be one in chat, Pangil.
No, not yet. Dr. Cha, please help me. No. 이거 채팅방 채팅방. Just a moment. Okay.
Okay, I will read them if you can't see them. One question
Okay.
is, have you looked at changes in splicing? Oh, wait, this is the old one. This is the old one.
Yes, first question. Yeah.
What is the sequence
I
space
got
around the
checked.
splice donor and acceptor sites that is used for the prediction? Let me repeat.
Yes, yes.
What is the sequence space around the splice donor and acceptor sites that is used for prediction?
So how long? So between acceptor and donor side?
Yeah, the sequence window.
I don't know. I'm sorry, I don't know. So just identify just AI splice AI algorithm. This algorithm, these programs provide some sites, so donor and acceptor site. So I don't know exact sites, so information. It's okay.
Thank you. And that question was from Reinhold Schaefer. Anyone else?
Yes, you.
Okay, so with that, Pangil, thank you very much for your presentation.
Okay, thank you very
interesting
much.
approach.
Okay.
Thank you.
Okay, bye-bye.
Okay, so with that, we will move to the last talk in this session prior to coffee break. And oops, I don't have my page.
Yeah,
but we're going to move from phospholipases to the products of phospholipases. And Dorothea Fiedler will be the speaker, and she will speak on dephosphorylation of inositol polyphosphates by the phosphatase MIN-PP1. Dorothea, the floor is yours.
Thank you. So I'm assuming you can see my slides and you can hear me well. Thank you to the organizers for this invitation. It's my pleasure to be here and present to you a really new and also really unfinished story, but I would love to get your feedback on our most recent work on this phosphatase MIN-PP1. And so since I haven't met most of you, just as a brief introduction, my lab is really interested in the inositol polyphosphates, the soluble inositol polyphosphates, so not the lipid anchored ones, and with a particular emphasis on these hyperphosphorylated molecules, the inositol pyrophosphates that carry 6, 7, or 8 phosphoryl groups. And since I am a chemist by training, we take a lot of different chemical approaches that would allow us to elucidate their signaling functions and their signaling mechanisms at the molecular level. But so for today, I want to take a little detour from the inositol pyrophosphates because, of course, throughout the years, we have been acutely aware that the inositol pyrophosphates must come from somewhere and that there's a really complex biosynthetic pathway that ultimately leads to the formation of IP6, which is shown down here, which serves as a precursor for most of the inositol pyrophosphates. And what I hope you can see here on this slide is that this whole biosynthetic machinery is it's a complex network of pathways and phosphorylation reactions. And what I hope you can also see is that there are way more arrows annotated that sort of go towards the end of this biosynthetic pathway. And when it comes to the dephosphorylation of these species, many things are actually not known. And this led us to start reading more about this one particular phosphatase called MINPP1, which to date is the only known phosphatase that can utilize IP5 and IP6 as a substrate and dephosphorylate these inositol phosphates. And just as a brief introduction to MINPP1, so this stands for multiple inositol phosphate phosphatase. It belongs to the histidine phosphatase family, and these phosphatases are sort of spread throughout all different areas of life. In plants, there are phytases that are important for releasing phosphate, and bacteria and many other organisms, these phosphatases exist as well. But as I said, in mammals, MINPP1 is the only phosphatase known that can utilize IP5 and also IP6 as substrates. And it sort of occupies as such a really central position in this large biosynthetic network. And what has most recently renewed, I think, many people's interest is were two separate studies that both showed that loss of function variants of MINPP1 were associated with pontocerebellar hyperplasia. And this is a rare neurodegenerative disease with prenatal onset. And so the groups of Vincent Cantangrel and Adolfo Sayardi also went on to show that loss of MINPP1 causes an early neuronal differentiation defect. And also this figure here is from their paper, where they showed that MINPP1 deficiency causes an imbalance of inositol polyphosphate metabolism. And so I'm seeing myself and lots of you in front of my figure. So I'm hoping I'm pointing into the right direction, but I think what you can see all the way over on the right is that IP6, for example, is significantly increased in cells lacking MINPP1. And it was suggested that this IP6 mediated chelation of metals could also be involved in disease pathology. But also other things are different in these MINPP1 knockout cells. So you can see there's an alternative IP5 species that accumulates, and these MINPP1 cells lack an IP2 and an IP1 species. And yeah, as I mentioned, sort of with renewed interest by these recent studies, but also with MINPP1 being a central phosphatase in this whole network of inositol phosphates, we felt we should take a closer look at this enzyme. And so those of you that study inositol phosphate metabolism are, I think, acutely aware that measuring these metabolites in complex samples is still a challenge. So the classical method is using the strong anion exchange HPLC method. That is also the data that I just showed you from Adolfo's lab, where they used tritiated inositol, and you can feed that to cells. It gets incorporated into the inositol phosphates, and then you do an extraction of the inositol phosphates, separate them by HPLC, and then look for radioactivity in your fractions. And it is a widely employed method. It's robust, it has high sensitivity, but of course you have to use radioactive material, and absolute quantification is not easy. Then also Adolfo's lab came up with this relatively simple method using PAGE to separate inositol phosphates based on their charge on a high density polyacrylamide gel. And this, again, is a much simpler setup. It doesn't require complex equipment, but also the quantification here depends on charge, and lower inositol phosphates are difficult to visualize. And then most recently, Henning Jessen's lab came up with a great new method using capillary electrophoresis coupled to mass spectrometry. And that really is another high sensitivity method with great resolving power. You can quantify via isotope standards. And so I think this is going to become more and more frequently used, but it is an expensive setup to buy. And for the lower inositol phosphates, such as IP1, 2, and 3 species, the resolution is not as good as we would like it to be. And then also what these methods, in our view, are often lacking is that you always require standards to definitively assign inositol phosphate species, and structural information is often missing. So what we wanted to do was to take an NMR-based approach using nuclear magnetic resonance and relying on 13C labeled inositols for metabolic labeling and subsequent analysis. And so two former students in the lab, Robert Hammel and Robert Puschmann, came up with a really nice and scalable synthesis of uniformly labeled myoinositol starting from 13C labeled glucose. And again, this was inspired by some of Adolfo's work, who had done something very similar on a smaller scale. So what we do here is we take 13C labeled glucose and phosphorylate it with hexokinase to generate glucose 6-phosphate. And this can then be isomerized by an inositol phosphate synthase to myoinositol 3-phosphate, which we subsequently dephosphorylate with alkaline phosphatase. And then we use a chemical derivatization and purification strategy to isolate our uniformly labeled myoinositol. And we get a really nice overall yield. It's about 50%. We can do this on a gram scale so that we really can obtain large quantities of 13C labeled myoinositol. And the two Roberts then went on to convert the myoinositol to IP6 and also IP5 using chemical methods, which was a relatively straightforward chemical synthesis. And they also further converted IP6 to 5-ppIP5 using the enzyme IP6Ka. And we then characterize these molecules using 2D NMR spectroscopy. So this is a 1H13C HMQC spectrum of IP6 that you can see down here. This is at 5 micromolar concentration, so a relatively low concentration. And I hope you can see is that we get distinct signals for the different CH groups at different positions around the myoinositol ring. There is a mirror plane in this molecule, so therefore these 1 and 3 and 4 and 6 positions are not distinguishable, but we get a very diagnostic spectrum. You can see this in particular if you look at the spectrum over here for IP5. So this is now lacking a phosphoryl group here at the hydroxyl group, and the signal for the 2 position has moved a lot when you look here and compare it to the IP6 spectrum. Yes, so we get diagnostic NMR spectra for the different inositol polyphosphate species, and so we can use them to assign which species we generate in solution. And we've done a lot of enzymatic assays at this point, where we can use our labeled substrates to monitor interconversion of IP6 to IP7, or in other scenarios with different kinases, other species. But here specifically, we work with Gabriel Schaaf's group, just to give you an example and characterize this plant protein called ITPK, which converts IP6 to 5-ppIP5. And we can monitor this in real time using NMR and extract kinetic constants, etc., from this data. So it is quite convenient when you work with purified enzymes. But it also turns out that this works for metabolic labeling, followed by analysis of the complex mixture. So we can feed 13C labeled inositol to mammalian cell lines. This gets incorporated into the phospholipids and the soluble inositol phosphates. And then we basically just do a lysis and record an NMR spectrum. And this is what these NMR spectra look like. There's a lot going on, but there's a characteristic region for inositol phosphates. If you zoom in, this is what this looks like. And one thing that I forgot to mention is that a lot of these, or all of the labeled peaks actually display this sort of triplet pattern. This is because you have adjacent 13C nuclei and they couple to each other. And we can then detect and also quantify amounts of IP6 and IP5 and also inositol pyrophosphates from these mixtures. One thing when we first recorded these spectra is we noted all of these other peaks that were not IP5, 6, or 5-ppIP5 but other inositol phosphate species that we could not assign back then when we published this paper. So we knew they had to be other inositol-derived species, presumably other inositol phosphates, but we didn't know what they were. And so this led a new student in the group, Min, to tackle this more systematically. And so he said, well, let's look at it in a systematic fashion. So here, as an example, I'm showing you the spectrum of IP3, so 145 IP3. And this is not symmetrical, so we get 6 different signals for the different CH groups here in this HMQC spectrum. And using different correlation spectroscopies, Min can assign them to specific positions. So we know this is the blue CH, the purple CH group here, and down here we have the turquoise CH group, and this is our assigned spectrum. So Min then repeated this exercise for 20 other inositol phosphates that we either bought or synthesized or obtained from colleagues, and recorded all these spectra. And while I know this is a lot of data to take in, I hope it is apparent that these signals cluster, right? So for this 2 position, for example, there's a whole group of CH groups that are up here, and then a whole other group of CH groups down here. And when we look at the assignment, we can see that the CH groups adjacent to hydroxyl groups will be the ones up here, and CH groups next to phosphoryl groups are in a different region of the spectrum. And so with this information, we have a pretty good idea of if we see a specific signal, whether it belongs to a phosphorylated CH or non-phosphorylated CH group, and which position it is attached at. And so we should also be able to assign new species based on all this information. Okay, with all of this information, Min went back and looked at some of our old labeling spectra. And again, this is what these 13C labeled spectra look like. And he can really assign every single peak in here at this point. I'm not putting arrows to all of them because it would get too complicated, but there are, of course, our old friends IP5 and IP6 here that we know very well. But then upon close analysis, he sees that these wild-type cell lysates, and this is not only true for these HEC293 cells, we've looked at different cell lines, and there's always a really significant amount of this IP2, so it's with the 1,2 phosphorylated pattern here, and then also IP1, which just carries a phosphoryl group at the 2 position. And so when we now look at a cell line, the corresponding cell line that's lacking MIN-PP1, we find that there are significant differences. And so as Adolfo already reported, there is a significant increase of IP6 in these knockout cells. And also, as was already seen in the radiolabeling traces, IP2 and IP1 are absent. So this is what used to be there and is now missing in blue here from the red spectrum. And then instead, we get a really large amount of a new IP5 species shown here in pink, which we know based on the chemical shifts is dephosphorylated at either the 1 or the 3 position. So there's a different IP5 species in here. And so the quantified values are over here in this diagram. And through some other experiments, we've really convinced ourselves that the formation of this IP2 and IP1 species are dependent on MIN-PP1 and the presence of IP6 in cells. And these knockout cells contain a new or uncommon version of IP5, which we presume is dephosphorylated at the 3 position. So this was all good and interesting. And then Min wanted to recapitulate this biochemically and see if we could make sense of this data in cells using a purified enzyme and monitoring the dephosphorylation sequences. So he spent a fair amount of time in optimizing the expression and purification of MIN-PP1 and got good enzyme, an active enzyme in hand. And then he basically incubated it with IP6 or IP5, the classical IP5 that is not phosphorylated at the 2 position, and monitored dephosphorylation over time, recording HMQ spectra at given time points. And then he can integrate these spectra and plot dephosphorylation sequences. And so what you can see at the top is the dephosphorylation sequence for IP5. It is relatively rapid, and we see formation of IP4 relatively quickly. And then over time, IP3 starts coming up, and this is really the species that dominates here for a long time. And this is IP3 with a 1,4,5 phosphorylation pattern, so the classical IP3. IP6 is a much worse substrate, so the dephosphorylation is relatively slow, but there is phosphatase activity at the 3 position first. So this is a 3OH IP5 that builds up, and then from there, sort of a complex dephosphorylation cascade kicks in. When we plot this out, maybe this is easier to look at. This is shown for IP6 down here. It goes with dephosphorylation at the 3 position, then yes, a lot of intermediates come up, but at the end, it merges on a common intermediate, and this is this 1,2 IP2, which we saw in cells, and also the 2-phosphorylated IP1 species, which is the cellulose species. And as I mentioned, IP5 sort of reverses the forward sequence from IPMK, so it takes IP5 back to IP4 to IP3, and then here it gets a little more complicated, but IP3 is the predominant dephosphorylation species that we detect here. But then the big question is, in these cell lysates, where does this IP5 species come from that was dephosphorylated 3 position when they're lacking MIN-PP1? That didn't seem to make any sense. So Min asked whether what's going on if we take lysates and add them to our inositol phosphate substrates. And so what he did is he cultured cells, and then either wild type or lacking MIN-PP1, he lysed with a mild buffer so that proteins would stay intact, and then incubated with IP5 or IP6 for 48 hours, and then did a titanium dioxide extraction and recorded the NMR spectra. And so for IP5 here, this is maybe easier to look at the beginning. You can see the wild type cell lysate completely dephosphorylated the IP5, so there's nothing left. We see buildup of IP3, just as we saw before in the biochemical experiment, and this is the predominant product here. If we have a lysate that is lacking MIN-PP1, that lysate doesn't know what to do with this IP5 here. So there's a little bit of 3-phosphatase activity, but this is pretty much it. IP6, also consistent with the biochemical characterization, is a much worse substrate for this lysate. So the wild-type lysate dephosphorylates IP6 to IP5, again, dephosphorylated at the 3 position. But interesting, also the MIN-PP1 knockout cell line still exhibits this 3-phosphatase activity. And so we think that this residual this alternative 3-phosphatase activity is what could be responsible for generating this intermediate and also for accumulating this intermediate in MIN-PP1 knockout cells, because MIN-PP1 then is missing to further dephosphorylate this intermediate. And so with this data, and I should say that these last sort of experiments are relatively recent, we've arrived at this overall scheme where we think MIN-PP1 actually plays multiple roles. So on the one hand, I think it's confirmed also with older data from Steve Shear's lab that IP5 here can be dephosphorylated by MIN-PP1 back to IP3. And in this way, MIN-PP1 here is involved in recycling higher inositol phosphates into this lower phosphorylated inositol phosphate pathway. But then what is interesting to us is that if IP6 is a substrate for MIN-PP1, it seems to put it on an alternative pathway that doesn't overlap with any of the other biosynthetic pathways. So it first takes off the phosphate at the 3 position and then phosphorylates it further down to this IP2 and IP1 species. And of course, we're now really curious to know, are these sort of end products or garbage products, or is there inositol recycling going on of some sort, or do these inositol phosphates have a specific function in cells, right? And we're now getting going towards looking at how dynamic are these pools of IP5 and IP6, and also these IP1 and 2 species with and without MinPP1. And so we're setting up pulse chase experiments right now with differently labeled inositol, exactly. And the other big question, of course, is what is this unknown phosphatase that we do believe is able to dephosphorylate here at the 3 position when MinPP1 is not there. And so, yes, as I told you, it was an unfinished story. We don't know the answers to any of this. This is what we're currently thinking about and where we're heading. I hope I was able to show you that this 13C labeling is a useful technique, especially when looking at the lower inositol phosphates. We've learned a lot about the structure of these different metabolites. We were also able to annotate these dephosphorylation sequences and found that the biochemical and also the cellular labeling data actually track quite nicely. We also think there must be another 3 phosphatase that is able to dephosphorylate IP6, and we would love to know what this phosphatase is, and then of course also know how this phosphatase interplays with MinPP1 in order to regulate this whole network for dephosphorylation sequences. And so with that, I would like to thank my group. I would like to thank the 2 Roberts, 2 former students, and then Min, who's again obscured by this bar with pictures of myself and other people. I think he's all the way over here wearing sort of a light green shirt and shorts. He did all of the work that I just told you about. Thank you also to our collaborators, Peter Schmiede, he's our NMR person at the Institute, always giving us great advice and help. Adolfo for providing us with the cell lines and also advice along the way. And then also Esther and Gabriel for our collaboration on the plant inositol phosphates. And then I would like to thank all of you for your attention, and I'd be happy to take any questions.
Thank you, Dorothea. So again, let's proceed this way. Let's consider chat first. There's a question up there. This is from Tatiana Igumanova. And her question is does generating cell lysates require D2O, full or partial, to suppress strong proton signals from water, since many of the diagnostic signals you are working on are in the 4 to 5 ppm region?
Yes, so the work that we've done with the cell lysates, we don't use D2O during the lysis or anything, but there is a titanium dioxide enrichment step. So that's how we get rid of most of the water, and then we resuspend our inositol phosphates. And yes, sometimes we get a strong water peak still, because when we resuspend after the enrichment, we do that in D2O. And sometimes we need an additional lyophilization and resuspension in D2O when we work with these isolated inositol phosphates.
So Dorothea, can you see the questions on chat?
Yeah, I'm trying to stop sharing and then that would be easier for me. Yes, okay, great, I can see them.
Okay, you know what? So I'll read it and then I have to turn off the microphone.
Okay, but I got it too. I can.
A second before the sound comes on. So Lois Weissman asks, can the analysis by NMR be adapted to measure phosphonostide polyphosphates that have been converted to the glycerol inositol polyphosphates?
Yes.
And the question there is that would allow you to determine PI45P2 is the sole connection between lipids and solids.
Okay, so I would like to talk to you about this more. So we started this project with one of my colleagues who is an expert in solid state NMR spectroscopy, and the idea was that we should be able to look at membranes and membrane phosphoinositides using NMR. This turned out to be quite challenging, and also the sensitivity of NMR is not as great as it is for radio labeling, for example. So there is still room for improvement. And so then your question regarding the glycerol inositol phosphates, that is something that I don't know all that much about. I can tell you that we see a lot of these GRO-Ps or GRO-PIs in the soluble inositol phosphate fraction, and we can quantify them well. And but what the connection is to PI45P2 is not clear to me. Maybe we can talk about this.
So I think I can help there.
Okay.
I think basically what she's asking is can you label cells with the C13 inositol, isolate the lipids, deacylate them, and then basically put the deacylated material into the NMR to do the assignments?
That would be, we have not tried that.
Is that right, Lois?
I think in principle that should be possible. Yes, thank you. But we would need multiple plates probably, but I don't see why we shouldn't be able to do it. We can see, we can say the phosphatidylinositols if we do a lipid extraction, right? So if we take our NMR samples and do a methanol chloroform extraction, we can see them by NMR. Yes.
Actually,
okay. And George,
before
I'm
you
going to
go,
jump in front of you very quickly because I have sort of a related question. But so there's the presumption that all of these inositol phosphate pools kind of come from the same bank account, that the phospholipases actually produce a common pool of IP3 and then they all emanate from there. And now my question is I know it's early days, but have you looked at PLC knockdowns, for example, in HEC2/3 cells to actually ask, is this really true? Are there perhaps pool specificities that are absent in these sort of other bulk analyses?
Yeah, that would, I mean, those are great questions. And those are questions that we would love to look at either by NMR, or we could also use the CEMS spectroscopy in combination with the 13C labeled inositols and also 13C labeled glucose, for example. I mean, Adolfo's lab recently showed that there is a PIP independent pathway for inositol phosphate synthesis in mammalian cells via ITPK. And yeah, I think a lot of questions when it comes to where are these pools localized. I mean, MinPP1 is supposed to be localized to the ER. How is this really working, right, as a global phosphatase for these inositol phosphate species? I think with new analytical methods, if we could look at subcellular fractionations, for example, that would be really helpful, right? Because unlike the phosphoinositides, we don't have good reporter domains that we can use to image things, for example, in cells, right? So I think, yes, it's going to be a combination of techniques. And we haven't looked at PLC knockouts, but I'm sure there is a phospholipase independent synthesis pathway. And of course, that also plays into our ideas about inositol phosphate recycling, or these sort of being shuttled onto different metabolic dephosphorylation pathways, and seeing what that does in certain cellular settings.
Okay, thank you. And George Carmen is asking, how much do you know about the enzymology of MinPP1? So I guess it's a pretty broad question. Quick answer, please.
We've started extracting reaction rates, doing kinetic modeling on IP5 and IP6 dephosphorylation, but it is tricky. That is my quick answer.
Very good. And Adolfo had a question, but I think you touched on it in your previous answer.
Yeah.
I think we'll,
I'd love to talk to Adolfo about this.
Let it go.
Yeah. Adolfo, we should talk.
Any other questions? Yeah. Okay, with that, 5-minute coffee break. Thank you.
Thank you.
Okay, so Lucio told me that people who are online can actually view some of the very nice pictures of the Academy. Oh, they're projecting them now.
Okay, we should start. So please have a seat here. And those that are join us virtually also have a seat. And I would like to thank again Lucio for this organization, and to let those people that are virtually only connected that they are missing a real experience to sit in a room that very unique. And with that, I would like to start and give the stage, the virtual stage, to Joel Pomeranz.
Hi, can you all hear me?
Yes, we hear you.
Okay, great. Thank you. Let me share my screen. Thank you very much for your attention. I really want to thank Lucio for the invitation and all of you for being here today. I'm going to talk about our recent studies on mechanistic impact of dominant negative CARD11 mutations in primary immunodeficiency. So for many years, we've been very interested in the signaling pathway downstream of antigen recognition in the adaptive immune system, downstream of recognizing antigen both by T cells and B cells by the TCRs and B cell receptors. This is a highly specific pathway. It's absolutely critical for the adaptive immune response. This is the signaling pathway, for example, that your coronavirus vaccines are eliciting. And we've been interested in what the molecular components are and how they function mechanistically. Our early connection to disease was the fact that this pathway is often dysregulated in leukemias and lymphomas that develop mutations that turn on the pathway without any antigen recognition. We've been focused for many years on the signaling hub, CARD11. It's a multi-domain adapter protein. It's absolutely required for signaling downstream of the T cell receptor and the B cell receptor to NF-kappa B, the JNK pathway, and the mTOR pathway. And basically, it functions as a signal responsive scaffold. The protein has evolved to receive signals from the engaged receptors on the surface of the B cells and T cells and turn that information into the assembly of a complex that activates NF-kappa B, mTOR, and JNK pathways. We know it's really important in human disease. It's first recognized as very important in many cancers of the immune system due to a wide variety of gain-of-function mutations associated with different forms of B cell and T cell lymphoma. And more recently, in the last 7 years or so, there have been many families described with primary immunodeficiency with germline mutations in CARD11. These are families that are highly susceptible to bacterial, fungal, and viral infections. And they segregate along different genetic patterns, but there are both gain of function and loss of function mutations of CARD11 in these families with primary immunodeficiency. For many years, we've been interested in, what makes this molecule, this multi-domain adapter protein, so susceptible to gain of function, loss of function mutations, dominant negative mutations that I'm going to talk about in a moment. How do these mutations affect biochemically the signaling? How do they cause or influence disease? And what do they reveal about normal antigen receptor signaling? Now, we know a lot over the years. We've learned a lot about how this protein functions. What I've shown on this slide is the CARD11 signaling cycle. In normal B cells and T cells, before the antigen is recognized, CARD11 is in this closed inactive state. It's basically folded upon itself, leaving protein-protein interaction domains not inaccessible to protein factors, and it's kept in this closed state through this inhibitory domain, which binds intermolecularly to different domains of the protein. And then when the receptors engage, when you see antigen, in this mysterious process, the protein opens. Once it opens, it can recruit cofactors in this cofactor association step, and there are quite a large variety of factors that are recruited to the protein, including BCL10, MALT1. MALT1 is a protease that many of you may have heard of. HOIP is an additional protein, and E3 ligase that also gets recruited. And subsequent to the recruitment of these factors, some of which are enzymes, a separate step activates the enzyme activity of some of these factors, leading to the production of signaling intermediates that go on to signal downstream to NF-kappa B, JNK, and mTOR. One of these critical intermediates is formed after the activation of this HOIP E3 kinase, which then modifies BCL10 with linear ubiquitin chains This intermediate then binds the IKK complex and then goes on to signal to NF-kappa B. So for many years, we've been studying this signaling cycle. We have mutants in the protein that allow us to identify these different steps in the signaling cycle because they prevent one or another of the pathway steps in the pathway. And we wanted to apply this knowledge to the understanding of how these mutations that are in these families with primary immune deficiency affect signaling. Now, there are many different forms, there are several different forms, I should say, of primary immunodeficiency formed by germline mutations in CARD11. One form is CARD11 deficiency, and this is caused by homozygous loss of function mutations. Basically, the patients don't have any functional CARD11, so their B cells and T cells can't activate in response to antigen, they can't fight off diseases. There's another form called Benta disease, which I won't talk about today, but this is caused by heterozygous gain of function mutations, which perturb B cell development in some ways, but in a lot of ways, this is still mysterious how these gain of function mutations block the immune system from responding. And then most recently, there have been recognized two forms of immunodeficiency that are accompanied by atopy. And atopy is a hyperallergic state, including food allergies, atopic dermatitis, hyper IgE production. And what was really interesting about these diseases, these families, is that these are loss of function mutations that manifest as heterozygous, and they appear to have dominant negative activity. And this is what I'm going to focus on today for the talk. You know, our initial introduction to the existence of this form of primary immunodeficiency was in collaboration with Chaim Roifman, who found a patient family in Canada and University of Toronto where the primary immunodeficiency syndrome segregating as an autosomal dominant fashion. These are all the affected individuals in this family were highly susceptible to bacterial, viral, and fungal infections. And this mapped to a single mutation in CARD11, this R30W mutation. And we wondered how could this mutation manifest as a heterozygote, as the dominant negative. So we performed this analysis. This is just an analysis of what the mutation does, either in the absence or presence of co-expressed wild-type protein. And basically what we did was we took the JERCAT T cell line that we have made deficient in CARD11 through CRISPR-Cas9 engineering. And here is a luciferase reporter assay that reads out NF-kappa B activation in response to cross-linking the T cell receptor with CD3, CD28 antibodies. CARD11 deficient cells, if you trigger them, they do not activate NF-kappa B, but if you rescue the cells and restore them with wild-type CARD11, they are completely competent to activate signaling, as shown here. This R30W mutation found in this patient family is completely dead. You know, we assayed it at equivalent protein levels of expression to the wild-type, and it's completely dead. But what was most interesting was that when we co-expressed this mutant protein with the wild-type protein, it completely blocked the ability of the co-expressed wild-type protein to signal, even though at the same level of expression, the wild-type protein is fully competent by itself to signal. And this was really the first simple indication that this protein was a really strong dominant negative. Sorry. Okay. So how do these loss of function dominant negative mutations work mechanistically? You know, there's this class of these loss of function mutations that can act this way as dominant negatives, but there's an entirely different class of loss of function mutations that only manifest with disease when they're homozygous. So what is the basis of this? What's the mechanistic difference? That's what we wanted to know. Why are some loss of function mutations dominant negative and others not? What step is affected? And another way to look at that is what signaling steps require all subunits of the CARD11 oligomer to be functional. Now, I originally represented CARD11 as a dimer in my simplistic schematic slides, but it's actually a higher order oligomer. We don't really know what the stoichiometry is, but we imagine, you know, you can imagine different stoichiometries. Here I pictured a hexamer. And in order to understand, you know, how a co-expression of a mutant and a wild type could affect signaling so drastically, you know, we sort of modeled it. We thought about it as a hexamer that once a mutant and wild type subunit is expressed in cells, it can sort into different oligomers with different combinations of wild type and mutant, and you can predict the relative frequencies of these different species simply by the binomial equation, which I won't go through. But you can imagine that if a mutant subunit, such as R30W, if it can affect the signaling of any oligomer in which it expressed and poison it, then you can achieve that type of level of inhibition that we see when you co-express wild type and mutant. So conceptually, this is how we're thinking about how a dominant negative could work. Using the binomial distribution, we can imagine through this that if, as the stoichiometry increases, the fraction of oligomers that are left active, which are the ones that are only populated by all wild type subunits, becomes less and less prevalent. Okay, so why are some of these loss of function mutations dominant negative and others not? How do they work? The approach that we took was to develop a panel of loss of function mutations and characterize their dominant negative activity and see if we can correlate a specific step in signaling that's affected by the strong dominant negatives that isn't affected as much by weak dominant negatives. So here you see a panel of CARD11 loss of function mutations that we developed in the lab as an offshoot of a different project. And this is the JERCAT T cell reconstitution assay where the cells lack CARD11. If you put in the wild type CARD11, you restore signaling to NF-kappa B. But all of these are functionally loss of function mutations. But what happens when you co-express each of these mutations with wild type? You know, what is their pattern of dominant negative activity? And what you can see here is that while they all looked identical to each other when expressed by themselves in cells, when you co-express them with wild type in this bottom panel here, they have very different dominant negative activities. These red guys here, when you co-express them with wild type, they severely block signaling that's emanating from the co-expressed wild type protein. These are moderately dominant negative, and these are weak. So we have the tools to which to dissect what is the step that's affected most by the strong dominant negatives and not by the weak dominant negatives. Which of these steps? Is this going to be this opening step? The cofactor association of, is there a particular cofactor that's going to be affected? Is it the activation of enzyme during the cycle? What is it going to be? So to make a long story short, we did a lot of cofactor association studies in the context of full length CARD11 just to see what these strong dominant negatives block. But first we wanted to analyze what these mutations block when they're expressed by themselves in cells. And not to go through all the pieces of data, we had a panel of mutants that were either strong or weak. The red guys are strong, the blue guys are weak. And when they expressed by themselves in JERCATs, all of these mutations blocked the association of all the cofactors that we analyzed, indicating that they probably block the opening step. And we could confirm this by taking a variant of CARD11 that we've generated in the lab that's constitutively open. Okay, so we can take this variant where we mutated portions of this inhibitory domain, which is usually responsible for keeping the protein closed and repressed. So this protein is constitutively open. It doesn't have to be opened by a signaling step. We can introduce mutations, these loss of function mutations, and show whether or not they affect the binding of cofactors in this opening step. And this is a very busy slide, but the point that I want to make is that for many of these mutations, even though they affected the binding of cofactors in the full length context of CARD11, they do not affect the binding of the cofactors once you mutate this inhibitory domain and artificially open the protein, meaning that these mutations are not at the protein-protein interface for recruiting these cofactors. They must be controlling the opening step. We then wanted to mix these mutant proteins with wild-type proteins in cells to see if there's a correlation. Do the strong dominant negatives affect the opening step? And do the weak ones not affect it so much? And that's exactly what we saw. You can see here some of these proteins that are starting to associate during this cofactor association step. They're only associating with the weak mutants, and they're not associating with the strong dominant negatives. So what we've concluded from this is that the dominant negative mutants poison the opening step, and that is the first critical step that these disease-associated mutations affect during signaling. That is why they manifest as heterozygous loss-of-function mutants in disease. This just shows it schematically. During normal signaling, you have the wild-type oligomer. It's shown as a dimer, but again, it's a higher order oligomer. When signaling happens, it opens. Then you associate cofactors, activate enzymes. If you express these mutants by themselves in cells, they fail to open when you engage the receptor. Their mutations somehow prevent the opening of the protein. And when you mix wild type and mutant subunits in cells, if you have a strong dominant negative mutant shown here expressed with the wild type, it prevents the whole oligomer from opening. Whereas the weak ones, you can get partial opening, partial association of cofactors, and somehow these mutations cause disease while these don't. Then we wanted to ask, can a dominant negative disrupt a different signaling step? So we had these panel of mutants, they all affected the opening step. The strong ones affected the opening more than the weak ones in the context of mixed oligomers, but can you also target a different step in signaling with the dominant negative? And for this, what we did was we used this constitutively open version of CARD11, and we basically mixed loss of function mutations with the otherwise wild type version of this constitutively open to see if even after the opening step, can a dominant negative mutation block signaling? And to make a long story short, not to go through the slide in great detail, but the answer was yes. So here you have these mutations in the context of the constitutively open protein. They block signaling as well. So they must not only be affecting the opening step, they must be affecting some other step downstream. And when you mix them with the wild type constitutively opening, here, for example, is this constitutively opening CARD11 at 1 concentration and at half the concentration. When you take this concentration of this constitutively open, otherwise wild type CARD11 and mix it with these mutants, you get drastic dominant negative inhibition. So there's clearly another step that is affected, that can be affected by these dominant negative mutants. So just to summarize, the dominant negative mutations that we have observed can disrupt the opening step in signaling. There's a second step following the opening step, which we're currently trying to elucidate to figure out what step that is that they can also affect. And what this says is that in these mixed oligomers, there must be coordination between the subunits of the oligomer during these steps. We've also obtained through this analysis, as an offshoot of this analysis, several mutations, several residues in the protein that are critically important for the opening step that are probably going to allow us to explore how this mysterious opening step actually occurs. And the main point here is that the deleterious effect of some of these loss of function mutants, but not others, on the opening step can partially be rescued by wild-type subunits in the mixed oligomer. Okay, so those are the weak dominant negatives. And that's why it explains why some are strong dominant negatives and some are weak. And this explains why some cause disease when heterozygous and others when homozygous. And I think I'm going to finish there and take any questions. I'd like to thank Jackie Bedsall in the lab, who's done the bulk of this work, and Chaim Roifman, collaborator, who led us to study this important primary immunodeficiency syndrome.
Thank you, Joel. We have questions here. Okay. You hear me also online? Do you
Yes.
hear me? All right.
Yeah.
So we have a question here in the lecture.
Hi, Joel. Great talk. Vitas Bankitis. Well, you have a lot of weak dominant negative mutants. And I'm just curious, is if you start mixing them, I know you can, this is a never ending game. Do you actually find interesting epistatic effects, you know, where they become either stronger or non-existent?
Oh,
So
that's a great question.
Mutants.
Yeah, that's a great question. We have been so focused on what the strong dominant negatives are blocking that we haven't attempted that analysis yet. And so it remains possible, as you suggest, that there is some epistasis. It also remains possible that some of the weak dominant negatives will actually complement each other and be affecting different steps, or even affecting the same step or the interaction with the same target with different affinities. But we don't know the answer to that yet.
Okay, maybe I can ask a question or two. So I guess there's no structure available.
That's correct. So there's the CAR domain, basically the first 100 amino acids, the structure of that is known, but the entire protein is about 1,100 amino acids, and all of the domains that I showed you that are important for this transition from closed to open, the structure is not known, and the precise oligomerization state is not yet known. And it's a very difficult protein to work with biochemically, by the way.
Yeah, but then how can you explain that most of the mutation which of course are localized slightly in a different position of the protein, cause the deficiency of function in the same way. Don't you think that there can be also alternative mechanism of some of these mutations that would cause eventually the lack of activity?
Oh yeah, definitely. So we have lots of different mutations that affect different steps in the pathway. So for example, we have mutations that were independently isolated that affect the binding of one cofactor versus another, for example. But what we did in this analysis was just to go blindly based on this first collection of loss of function and see, which are going to be strong dominant negative versus the weak and why, and they seem to affect the opening step. But you're absolutely right. There are lots of other mutations that affect some of these other steps, and we don't know for sure whether they are dominant negative or how strong the dominant negative activity of those is coming from analysis of a very specific step. There's also this other step after opening, which some of the strong dominant negatives also affect, which could be affecting some of those other steps, and we just don't know yet. We're still doing those experiments.
And my last maybe comment question is, have you tried to use AlphaFold to get the structure of the protein?
Yes, we have, and it's a complete failure because the most interesting portions of the protein are not structured by that analysis, and it doesn't assemble the protein into oligomers at all. So we think that a better attempt at the AlphaFold will be necessary, but we were hoping at some point to get structure in collaboration with structural biologists.
Okay, thank you. Then I do not see any question in the chat, so let's thank Joel again. Thank you very much.
Thank you.
And our next speaker is Stefan Schurman. I hope I pronounce it correctly.
Good afternoon, everybody. Do you hear me?
Yes, we hear you. The floor is yours.
Okay, I will share my screen. Oops. So do you see my screen?
Yes.
Thank you. Somebody is there. So good afternoon from Brussels, Belgium. I would like to thank Professor Lucio Coco and his team for the invitation and for organizing this meeting. Last year, I told you that we are interested by the analysis of the 5-phosphatase ENPP5K in the hematopoietic system. ENPP5K is a member of the 5-phosphatase family. Its structure is very simple. It's composed of the catalytic unit, which is active on PIP245 and on PIP3, and a stitch domain, which is responsible for protein-protein interactions and for subcellular localization. There are mutations in the human gene which are responsible. These mutations first are not fully inactivating. They are point mutations which replace one amino acid by another. And this mutation leads to an autosomal recessive muscular dystrophy, which is associated with cataracts, short stature, and intellectual disability. In 2008, if I remember well, Takenawa published the first knockout mice for this gene, and the full knockout are embryonic lethal at embryonic day 11, and the heterozygote are hypersensitive to insulin. And so we decided to analyze the role of ENPP5K in hematopoietic cells, and we produced in ENPP5K flox VAF-Cre mice, and these mice direct the expression of the Cre recombinase specifically in the hematopoietic cells. So the system is working. You have here a Western blot showing that the ENPP5K is absent in blood leukocyte from the knockout. And here by fax that the protein ENPP5K is absent from the bone marrow B cells. We started the analysis of these mice by looking at the blood. And as you see here, there was a very significant decrease in the leukocyte concentration in the blood of these mice. And this was particularly due to the nearly total absence of lymphocyte in the blood of these mice. But for some other white cells, there was an increase, like for basophils or neutrophils or monocytes. So we further analyzed these lymphocytes and we saw that B cells, CD19 positive B cells are severely decreased, as well as all the subset of T cells, CD4, CD8, CD3. And remarkably, in the blood of these mice, there was nearly no immunoglobulin, no IgG, IgM, or IgA. Since there are big alterations in the B cell lineage, we decided to focus our analysis on the B cell lineage. As you certainly know, the common lymphocyte precursor, we give rise to T cell and B cells, enter the B cell lineage, and in the bone, this is in the bone marrow, and differentiation occur in the bone marrow. The immature B cells then move to the spleen where they finish their maturation, T1, T2, and then mature B cells, follicular cells, marginal zone B cells, which when they encounter an antigen, the specific antigen can differentiate in plasma cells and secrete the antibody. This differentiation is controlled mainly by 4 receptors, interleukin-7 receptor, which control an essential transcription factor, PAX5, which is responsible for the commitment of the precursors, the progenitors in the B cell lineage, the pre-B cell receptor, the B cell receptor, and finally the B cell receptor in the presence of antigen, which trigger the differentiation of plasma cells and immunoglobulin production. We have found in our mice alteration at all this stage, but I will today only focus on the first stage, which is the commitment of the progenitor in the B cell lineage. So these are the first stage in the B cell lineage, fraction A and fraction B. As you see here, there is a partial blockade between fraction A and fraction B. Blue means wild type. And because there is an increase, a significant increase of cells in the fraction A of the knockout and significantly decreased in fraction B. So spontaneously, we went to this famous transcription factor, PAX5, which is essential for the commitment in the B cells. And we know from other studies in the literature that when you inactivate PAX5, there is a nearly complete block between fraction A and fraction B. And these cells in fraction A are able to de-differentiate in other cells, like in T cells, in macrophages, and in neutrophils. We saw that the expression by fax of PAX5 in the knockout cell was significantly decreased, meaning that there is a problem with the interleukin-7 receptor signaling in the absence of ENPP5K. So this is the receptor, which comprise two chains, the alpha chain, which is specific for interleukin-7 and which recognize interleukin-7, the gamma command chain, which is common to many other cytokine receptor, their transmembrane segment and cytoplasmic domain, with the JAK1, which is constitutively binding to the alpha chain, and JAK3 constitutively binding to the gamma command chain. So I told you that we have B cell differentiation defects, significant decrease in PAX5 expression in the absence of ENPP5K, and upstream we have STAT5, and as you can see here, there is a significant decrease of phospho-STAT5 in the absence of ENPP5K. So one step earlier, upstream, we have JAK1, phospho-JAK1 and phospho-JAK3. And I present only the result for phospho-JAK1, but we have exactly the same result for phospho-JAK3. There is also a significant decrease of phosphorylation of JAK1 when ENPP5K is absent. So this means that in the absence of ENPP5K, the early steps of the interleukin-7 receptor the activation are abnormal. We know that dynamic structure, we know from the literature that dynamic structure of the interleukin-7 receptor is essential for the activation. When interleukin-7 is present, it brings together the alpha and the gamma chain, brings together or closer the transmembrane segment and also the cytoplasmic domains. So JAK1 is more proximal to JAK3. They transphosphorylate, JAK1 phosphorylate JAK3 and JAK3 JAK1. And these lead to the activation of the receptor and the phosphorylation of STAT5. So given that we thought that the early step of the interleukin-7 receptor activation were abnormal in the absence of ENPP5K, we decided to use an indirect FRET assay in primary bone marrow B cells isolated from wild type and knockout mice. And this is following the method which has been described by Guala in 2018. In this indirect FRET assay, the fluorophore are not fused with the protein of interest. They are on the antibodies which recognize specifically the proteins. And we designed two types of indirect FRET, one which recognize the alpha and the gamma ectodomains, and one which recognize the JAK1 and the JAK3 kinase inside the cells. I must say that all the protein that we have targeted have the same expression. There is no difference between knockout and control mice. And so we stimulate one fluorophore, energy is transmitted to the second, and there is emission that we measure. And the measure of the emission is, of course, in direct inverse correlation between the distance of the two fluorophores, meaning the two antibodies, meaning the two proteins. But we had to validate this system. And first, we decided to define the minimal energy transfer by using an antibody which recognized JAK1, which is present at the cytoplasmic domain of the alpha chain, meaning at the plasma membrane, and PAX5, which is present in the nucleus. So the distance is very high, and as you see here, the energy transfer is null. The second control that we used is to use two antibodies which recognize the same target, JAK1. Here, the proximity is very high, and so the energy transfer is expected to be very high too. And this is the case. The third control that we use is to try to reproduce what is known in the literature concerning the ectodomains and the presence or the absence of interleukin-7. You see here that, as expected, and reproducing the result of the literature, when we add interleukin-7, there is an increase, a significant increase in energy transfer, meaning that the two ectodomains are closer when we add interleukin-7. And the last control that we did is the same for the two JAKs. We know from the literature that adding interleukin-7 results in a shorter distance between the two JAKs, meaning an increase in energy transfer. And this was the case, a very significant increase in energy transfer when we add interleukin-7. So we started to compare knockout and control cells. And interestingly, using the antibodies directed against the ectodomains, we found that there is a significant decrease in energy transfer in the cells in the absence of interleukin-7 and ENPP5K, meaning that the distance between the ectodomains in the knockout cells is bigger. So what happens when we add interleukin-7? There is a significant increase in energy transfer in the knockout cells, meaning that they are close together, but not still as in the control cells. So there is a problem in the dynamic structure of the ectodomains of the interleukin-7 receptor in the absence of ENPP5K. We did the same for the two JAKs, and surprisingly here, we found that the energy transfer is significantly bigger in the absence of ENPP5K in interleukin-7. So in basal condition, JAK1 is closer than the distance between JAK1 and JAK3 is smaller, is shorter in the absence of ENPP5K in the basal condition. And what was most surprising for us is that addition of interleukin-7 did not change at all the distance between the two JAKs when ENPP5K is absent, meaning that the distance between the two JAKs seems frozen at an intermediate between the basal condition and the stimulated condition of the control cells, but it's frozen and the distance does not change if we add interleukin-7, meaning that this distance seems to be insensitive to interleukin-7. So we decided to investigate the mechanism, and the first thing that we did is to look at the presence of ENPP5K in a complex with the alpha chain. Why? Because in the literature, it is known that ENPP5K and the alpha chain of the interleukin-7 receptor here have a common interactors, which is GRP78. So we decided to immunoprecipitate the interleukin-7 receptor alpha chain, and we found by Western blot ENPP5K. And inversely, we immunoprecipitate ENPP5K and we found the alpha chain of the receptor. So we are currently analyzing in more details the interaction between these two proteins. Is it direct, indirect? Does it need GRP78? And what are the domains which are involved in this interaction? Second, we decided to analyze the two substrates of the enzyme, and we found no difference in PIP3. In fact, we found no difference in the phosphorylation of AKT between knockout and control cells. By contrast, when we were analyzing PIP2 level, we found a significant increase of PIP245 in both fractions A and B of the knockout cells. So how can we reconciliate this increase in PIP2 level and the alteration in the structure, in the dynamic structure of the interleukin-7 receptor? And the solution is maybe, possibly, ionic interaction between PIP2, which is negatively charged, and positive charge in the interleukin-7 receptor. This kind of mechanism has been described with other receptors like the EGF receptor, the CD3 epsilon, CD3 zeta of the T-cell receptor complex, and for surface IgG. And thus, we were looking for such polybasic, positively charged amino acid sequence, and we found nothing interesting in the gamma chain. By contrast, here in the alpha chain, we found a very interesting amino acid segment. You have the transmembrane segment here in green, and you have just close to the transmembrane a very interesting sequence. And this sequence fulfills all the 4 criteria which have been defined by Chen-Ki Xu for the interaction of a peptide with the plasma membrane and for negative charge in the plasma membrane. And these 4 criteria is that the amino acid sequence must be close to the transmembrane and the membrane, which is the case. There must be several clusters of positively charged amino acid. There is 1 cluster here, a second here, and a third here, which are separated by hydrophobic amino acid, which is the case in blue. And finally, the fourth criteria is that there must be not too much negatively charged amino acid like D and E, which are here. So we are now collaborating with Chen-Ki Xu, who is trying to demonstrate or not that this peptide is able to bind to liposomes, which mimic the composition of the inner leaflet of the plasma membrane, plus minus different concentration of PIP2. Importantly, this segment is very conserved between species, and it's probably the reflection of an important role in of this segment. So we decided to analyze the proximity between the alpha chain here and the plasma membrane, because it's likely a reflection of the strength of the ionic interaction between PIP2, present here, and between the positive charge which are present at the juxtamembrane region in the cytoplasmic domain of the alpha chain. And so we found, interestingly, that in the absence of interleukin-7, there was an increased energy transfer, meaning that the alpha chain is closer to the plasma membrane in the absence of interleukin-7 when ENPP5K is absent. If we add interleukin-7, again, like for the JAK1, JAK3 distance, the distance between the alpha chain and the plasma membrane is exactly the same. There is no difference, on the contrary to control cells, where there is a smaller distance between the alpha chain and the plasma membrane when we add interleukin-7. So again, there is an insensitivity to EL7, and there is a freezing of the distance between the alpha chain and the plasma membrane. In conclusion, I try to convince you that the lipid 5-phosphatase ENPP5K interact with the alpha chain of the interleukin-7 receptor, that inactivation of ENPP5K leads to increased PIP2 level, probably to increased ionic interaction between PIP2 in the plasma membrane and the positively charged polybasic amino acid sequence in the cytoplasmic domain of the alpha chain. And this leads to alteration in the dynamic structure of the receptor, especially the alpha chain, the distance between JAK1 and JAK3, and more generally of the interleukin-7 receptor. This receptor, in the absence of ENPP5K, does not seem to respond properly to interleukin-7. And all these dynamic structure alteration probably are responsible for the alteration in interleukin-7 signaling and the B-cell differentiation in the bone marrow that we see in the knockout mice. So based on these results we obtained in on the wild type interleukin-7 receptor, we have decided to analyze the effect of ENPP5K inactivation on mutated oncogenic interleukin-7 receptor alpha chain, which are present in some patients who developed B and T cell acute lymphocytic leukemia. This mutant, in fact, in orange here, are present at the junction between the extracellular domain and the transmembrane domain. And they confer either a constitutive activation of the receptor or an hyperresponsiveness to interleukin-7. And we will start soon, very soon, this experiment by inactivating ENPP5K in the cell line that express different oncogenic mutant of this alpha chain. So nearly all the results that that I have presented today have been generated in the lab by Bastien Mous, who is a PhD student who will pass his thesis in a few weeks and is looking for a postdoc position abroad in immunology from 1st of January 2023. And I would like to thank him. He's really excellent, I could say outstanding. He has gold in his hand and gold in his brain. It's always important. I would like to thank Professor Chenki Su for the collaboration to try to know if this peptide sequence is able to interact with the plasma membrane and with PIP2. And finally, I thank Professor Barata and Israeli for providing my lab with cell lines which express mutated oncogenic interleukin-7 receptor alpha chain. Thank you very much for your attention. I'm ready for the question.
Thank you, Stefan. Maybe I can start. So I actually did not understand your model because you say that PIP2 play a major role in the dimerization, essentially, that is needed for the 2 receptor to be active. And you said, or you mentioned that this is maybe similar to other receptor like EGFR. But in fact, EGFR, you can get a very functional dimerization without even a membrane. So you can use an artificial membrane, you can use artificial detergent, and you'll get beautiful dimerization. You can even take only the extracellular domain and you'll get
yes.
Yeah.
Yes, maybe I was not very precise. I don't speak about dimerization. I speak about the cytoplasmic domain, which is able to bind to the plasma membrane. And for the EGF receptor, for example, there is a polybasic amino acid sequence which interact with the plasma membrane and which is responsible for a kind of decreased signaling, but it does not alter the dimerization. So it's really the conformation, the structure of the cytoplasmic domain without altering the dimerization. Okay? And we have the same with the CD3. In fact, in CD3, how can I say that? The cytoplasmic domain is hidden in the plasma membrane. And there are some tyrosine which are very important, which are hidden from the kinase, and the kinase cannot have access because the tyrosine and the segment of the cytoplasmic domain is within the plasma membrane, so unavailable for the kinase.
Hi, Stefan. A beautiful talk. This is Vitas. So I'll have a comment for you that I'll send by email suggesting an experiment, but here's one thought. So you have a fret, half of the fret pair that is actually on the cytoplasmic tail side, right? And the question is if you found a reasonable fret pair that you can incorporate into the membrane to actually do the proximity analysis that way. You're predicting that actually with the PIP2 binding, you're getting a differential distance from the membrane. in one case versus the other. So if you had a FRET acceptor or donor in the membrane, you should be able to test that. And I was just wondering if you've tried.
I think I presented that. I will show you. Do you see my screen?
No, not yet.
No.
No, we don't see your screen.
So I've presented an indirect FRET assay where we used a membrane probe, DEO, which is labeled with the fluorophore. So this probe marked the plasma membrane in green. And we use it, this first fluorochrome, plasma membrane, because it's a membrane probe, with an antibody which recognized the alpha chain. And so we could relatively measure the distance between the plasma membrane and between the alpha chain. And we saw that there are differences. And there is also a freezing of this distance, which is insensitive to interleukin-7. Maybe I was too quick on that, but we have this membrane probe in conjunction with the antibody which recognized the alpha chain. So we have done that. I think, if I understand your question, we have done the experiment.
Okay, thank you. I think for the sake of time, we have to continue.
Okay.
Our next.
Thank you very much.
Our next, yeah. Well, the only thing I can do is, yes. So our next speaker is Reinhold Schäfer. Please, Reinhold, the stage is yours.
Yeah, good afternoon. While I'm trying to show my screen, I take the opportunity to thank Lucio and his team for the great preparation of this meeting and his immense patience of bringing us here during the pandemic, which hopefully is winding down now. And I'm quite optimistic that the COVID restrictions will no longer be an issue next fall, especially because Italy is the European champion of vaccination of its population. So congratulations to this, particularly when I think about my own country. I would like to take you onto a trip into, let's say, traditional tumor cell biology. And my motivation is the following. To date, we understand the pathological function of many oncogenes and tumor suppressor genes pretty well. However, their overall number remains a continuous challenge. The COSMIC database in issue 94, for example, lists 567 driver cancer genes and tumor suppressor genes. In addition, there are 144 less well-characterized genes with similar function. Some driver genes, of course, stand out because of their high prevalence in tumors. For example, the KRAS gene is mutated in 95% of all pancreatic adenocarcinomas. Such genes, of course, have been characterized in much detail. But in view of the total number, do we really understand the function of all of these genes? The challenge of providing a functional understanding of cancer-associated genes, cancer driver genes, becomes even bigger when we consider the magnitude and complexity of tumor transcriptomes. I'm sorry, there's something wrong here. Of tumor transcriptomes. For example, if you look at the pathology atlas of the human transcriptome, listing profiles of 8,000 patients and 17 different tumor entities. And even if one tries to simplify the material under investigation in a kind of also traditional reductionist manner, and looks, for example, at the transcriptional alterations that are caused by a single cancer gene, in this case, mutant RAS, which is kind of the workhorse in my lab, one is confronted with a multitude of up and down regulated genes whose specific functions would have to be questioned. This does not mean, of course, that one cannot obtain valuable information about the role of deregulated genes in tumors. For example, here you see this Kaplan-Meier plot, which shows the role of an upregulated transcription factor as a marker for the demonstrated poor survival of the patient group. However, this information is essentially correlative and does not imply causalities. As I said, to study the tumor biological function of large numbers of genes, of less well-characterized candidate genes, is a major challenge. And then the following, I'm going to show examples of the analysis of potential oncogenes and tumor suppressor genes by means of transfer into or genetic manipulation of suitable, carefully selected recipient cells, followed by the observation of phenotypic consequences. And that is the principal workflow for both types of genes. It is based on lentiviral gene transfer and transient transfection or stable transduction of siRNAs into appropriate recipient cells. These are either spontaneously immortalized or, following introduction of telomerase, are genetically engineered to lose p53 tumor suppressor and RB tumor suppressor functions. This is important because in this case, an oncogenic challenge does not drive them into premature senescence or apoptosis. Now, as an example, I'm going to show characterization of SHIP2 PTPN11 mutants, which are associated with congenital developmental disorders called rasopathies. And you see the three examples here. This is Noonan syndrome, Costello syndrome, or CFC syndrome. The mutations in these syndromes affect different components of the RAS signaling system. And they occur also with low prevalence in hematological and solid tumors. Now, expression of SHIP2 mutants in pre-neoplastic cells, the ones that I showed before, leads to an increase in cell density, similar to that of classical RAS oncogenes. During this process, the cells change. They appear elongated, and this is quite similar to the effects achieved by introducing mutated RAS into these pre-neoplastic cells. Cellular transformation can, of course, then be quantified by measuring the length of cells, like here. And this, again, reflects a typical readout following the expression of mutant RAS genes. You can also monitor the strength of the transforming ability. Moreover, the mutants also induce the anchorage-independent proliferation phenotype, which is closely linked to tumorigenicity. Not absolutely linked, but very closely linked to tumorigenicity. Here are the colonies, the soft R colonies in the original, and this is the quantification of the number of colonies formed in soft agar following the introduction of these mutants. We have also xenotransplanted cells transduced with 2 of the SHIP2 mutants and wild-type SHIP into immunodeficient mice. We saw an unequal potential for tumor formation, which is shown here and here. The SHIP2 T468M mutant promoted neoangiogenesis and formed larger tumors. Immunohistochemical staining of the tumor sections from these xenotransplanted tumors showed PCAM1 positivity, which again indicates the onset of neovascularization. Accordingly, MAP kinase activity measured by Western blot in cell extracts from cultured cells and also from the xenotransplanted tumors is slightly increased, suggesting chronic pathway activation. So we conclude that SHIP2 mutants may act as typical oncogenes in the functional assays, although in some cases with weaker activity than the classical, for example, RAS or BRAF driver genes. Now, what about the characterization of genes whose expression is lost in experimental and human cancer cells? Actually, we have been studying RAS pathway responsive genes in the lab for many years. Actually, we published the first RAS-dependent genome-wide gene signature already in 2000, and numerous signatures with hundreds of genes have been published since then. The number of downregulated genes may even exceed that of the upregulated ones. This, of course, causes a need for prioritizing such genes for further functional analysis. And here is our attempt to identify robustly downregulated genes. First, we compare the transcriptional profiles of different cell types, mesenchymal versus epithelial, which express mutant RAS. And secondly, we focus on alterations conserved in evolution by comparing human, rat, and mouse cells, again, comparing normal versus RAS transformed cells. And please note that we used a customized microarray platform, which was specifically designed by us for interrogating RAS responsive genes. And we identified 4 robustly downregulated genes, which I refer to as RRD, RAS responsive down. The 4 RRD genes encode lysyl oxidase, cysteine-rich protein 61, folacetylide 1, and thrombospondin 1. We confirmed their negative regulation by individual analysis of their RNA and protein products, again, comparing normal versus RAS transformed cells. Now, in terms of gene function, in terms of transformation, the question now is whether their suppression is just a secondary consequence of RAS pathway activation, or is it causally involved in any way? To address this question, we silenced RRD gene expression by introducing siRNAs into pre-neoplastic human fibroblasts. These are the ones here. Transient silencing of RRD gene resulted in morphological conversion of cells, as is depicted here, which was indistinguishable from the effects of introducing mutant RAS. Moreover, we saw that RRD gene knockdown conferred the ability to proliferate on polyhema-coated plates that prevent cell attachment. So this is shown here by these high bars. The gain of anchorage-independent proliferation was accompanied by activation of the MEP kinase pathway, as measured by increased phosphorylation of the signaling kinase ERK1 and 2 that you can see here, here, and here. Now, to rule out that we are only seeing transient phenotypic effects here, we introduced LOX short hairpin RNA controlled by a lentiviral vector into the same pre-neoplastic cells in order to achieve a stable LOX knockdown. You can appreciate the typical elongation of cells shown here, as well as the induction of soft agar growth, although the colonies were significantly smaller than those induced by RAS. This suggests that either stable silencing of a single RRD gene, in this case LOX, is not sufficient for cellular transformation, at least during prolonged time in culture, or alternatively, that cellular adaptive mechanisms impair the ultimate expression of the transformed phenotype. And by the way, we see similar effects of stable LOX silencing in experiments with pre-neoplastic rat cells. So again, the question is that effect conserved in evolution? And this seems to be the case. Now, if we accept that silencing of LOX in pre-neoplastic cells generates the transformed phenotype, the function of the gene indeed corresponds to that of a transformation suppressor that normally protects from neoplasia rather than induces it. To prove this, we expressed LOX in RAS transformed LOX negative cells. You see here that the RAS induced neoplastic phenotype, this is normal, this is RAS transformed, is almost completely reverted. And the cells also lose their ability to proliferate under soft agar conditions, under conditions of anchorage independence here or here. The reversion of the transformed phenotype was particularly strong in LOX in single LOX expressing clones, but it was weaker in cell populations, suggesting that subpopulations of LOX negative cells were enriched during prolonged cultivation. Now, looking again at MEP kinase activity, the modulation of stable LOX expression in RAS transformed cells here, seen on the left, these are the 2 bars here, had a negative impact on MAP kinase activity, while the reverse experiment, the stable silencing of LOX in pre-neoplastic cells, stimulated MAP kinase activity, suggesting that the level of LOX definitely controls MAP kinase activity, most likely in an indirect way, and supported its role as a suppressor of RAS-mediated neoplasia. Interestingly, we also saw modulation of RAS expression itself. So you would expect that also the downstream pathway is blocked in some way. We assume that this is due to the fact that LOX interferes with the feedback regulation of the RAS-MAP kinase pathway that involves interaction between the receptor downstream signaling kinases and even effects of transcriptional targets, particularly MAP kinase pathway transcriptional targets. The mechanism of LOX activity as a tumor suppressor and its modulation of MAP kinase activity are not fully understood. There's previous work by Jay et al. That has suggested a role for LOX in cellular transformation. Another paper by Finko et al. Provided evidence for the functional relationship of RAS and NF-kappa B signaling pathways, in that it not only takes two to tango, but also two pathways to achieve full transformation. The possible role of LOX in this crosstalk requires its post-translational processing. Proteolytic cleavage of the LOX proenzyme produces the secreted enzyme, which is secreted and stabilizes the extracellular matrix, basically by oxidative deamination of peptidolysins in procollagen and proelastin. The second cleavage product is a 80 kilodalton polypeptide that can prevent the localization of PDK to the membrane, and in this way inhibit activation of the upstream kinases that phosphorylate the NF-kappa B inhibitor I-kappa B. I-kappa B normally sequesters NF-kappa B in the cytoplasm and keeps it inactive. Only if I-kappa B is phosphorylated, the inhibitor is subjected to degradation via the proteasome. In this case, NF-kappa B can translocate to the nucleus and activate gene transcription, like here. Therefore, we conclude that suppression of LOX expression by oncogenic RAS signaling and the absence of the polypeptide is a prerequisite for effective crosstalk between the two pathways, MAP kinase pathway and NF-kappa B pathway. In contrast, expression of the polypeptide in normal cells keeps the NF-kappa B branch of the crosstalk inactive and hinders effective cellular transformation or protects from cellular transformation. It is clear, however, that several lines of evidence, which I don't have the time to report here, have suggested that LOX interactions are manifold with other cancer-related signaling proteins as well, particularly with receptor tyrosine kinase, this membrane-bound receptor tyrosine kinases, and also with TGF-beta. Now, this is my summary. The traditional cellular transformation assays, which I have kind of revisited here, are suitable for systematic phenotypic analysis of candidate oncogenes and tumor suppressor genes. Commercially available tools, these are very important if one thinks about multiple analyses, which are synthetic cDNAs, expression vectors, and commercially available as HRNAs or vectors controlling short hairpin RNAs, facilitate the screening of multiple candidates. These assays are scalable for a medium throughput. I think it is clear that gene transfer assays indicated phenotypic conversion motivate a plethora of downstream analyses, which are more oriented towards the mechanism or understanding of how these genes affect the transformed phenotypes. I showed evidence that RASopathy-associated SHP2 mutants possess oncogenic activity similar to classical oncogenes like RAS and BRAF. We describe several RRDs, this is a misprint here, RRDs, RAS-responsive down genes that suppress the cellular transformation and modulate MAP kinase pathway activity. There is evidence that the LOX peptide can interfere with the crosstalk between MAP kinase and NF-kappa B pathway that is essential for efficient neoplastic transformation, but as I mentioned, there's evidence for multiple interactions between LOX and other signaling molecules. In the lab, we made an effort using tandem affinity purification, also yeast to hybrid screening, and we found some evidence for an interaction between LOX and heat shock proteins, which are known again to interact with BRAF and other RAF isoforms. That could also at least give an indication for an interesting mechanism of how LOX modifies the MAP kinase pathway. But this is currently speculative from my side, and the data in the lab are not that worked well out so far. Now, there's limitation for these assays. Obviously, the proliferation and genetic programs of our cell models are mainly controlled by the RAS-MAP kinase pathway. So it's not clear if the effects of other pathways, just to mention the Wnt signaling pathway, Notch and Hippo pathways, may not be captured by similar assays. These would perhaps require different types of recipient cells. The candidate oncogenes that we looked at are expressed ectopically to a very high level, sometimes to a medium level, but still overexpressed. And such experiments, of course, would need validation in isogenic model systems or the application of more refined methods for genetic manipulation, just to mention knockout experiments or knock-in experiments using CRISPR technologies. Everything that was observed in cell culture, of course, requires in vivo validation. And let's say even looking or thinking about human model systems, one would even utilize patient tumor-derived organoid cultures, which are also easy to manipulate. I would like to give credit to my collaborators. Part of the work that I've presented was done by 4 graduate students in the lab, Minkam Hati, Anja Wieland, Shalu, and Paula Medina Perez. I would also like to thank my former colleagues in the lab, Professor Christine Seers, Dr. Oleg Czernitsa. Our work was funded by the National Genome Research Network, by the Federal Ministry for Education and Research. And I hope I was able to convince you that phenotypes matter in biology. And if you compare the biology business with the real estate business, I think it is clear that in real estate business, the first criteria for success is location. The second one is location, and the third one is location. And in biology, I think it's 1, 2, and 3 phenotype. Thank you very much, and I'm happy to take questions.
Thank you very much, Reinhold. We have here a question.
This was a beautiful study of non-canonical pathway to affect the NF-kappa B. Yeah. Have you checked the canonical path? Can you hear me?
Yes, I can hear you. I can hear you.
Yeah, canonical NF-kappa B activity in such situation.
Yeah, we see no major difference in when you use EMSA assays. We haven't seen any difference between the normal and the transformed cells, at least in the human fibroblasts that are presented. So the interesting thing about this interaction of the LOX peptide with the NF-kappa B signaling is that, to my knowledge, it is kind of largely neglected by the NF-kappa B science community. So one of my colleagues here in Berlin, who's working at the Max Delbrück Center for Molecular Medicine in Buch by the name of Klaus Scheidreit, also told me that this could be interesting to follow up. But so far, there's not much work devoted to kind of confirming this in different model systems, in different cell types, for example. But at least there's a way to approach this question using, perhaps it would be interesting or maybe important to purify the peptide and then do experiments along these lines. Of course, we have no control in our cell systems how efficient the proteolytic cleavage out of the proenzyme will be.
Okay, thank you. Any additional questions? Well, I don't see here and I don't see in the chat. Then I have to conclude that I fully agree with you, Reinhold. Sitting in this location, I would say that location is really the most important issue.
Absolutely. Yeah, I would have loved to be there.
Okay. So let's thank again Reinhold for this. And our next talk is here. It's Mina that will, I don't know how this is going to work, but to you, and I give you the stage.
Thank you. Yes. Thank you, Lucio, for having me here. After your excellent talk, I don't feel like giving my talk. Anyhow, I would talk about the mTOR pathway in glioblastoma. So I would not talk about background. Anyhow, glioblastoma falls into two categories, primary and secondary. Primary is generally occurs de novo, and secondary is from the primary tumor progress to the real glioblastoma. Both of them are dichotomized by the age of occurrence. Primary is an older age than the secondary one, but they have a certain gene abnormality which is common. However, the percentage of changes are very different. And also the secondary GBM has a P53 abnormality and IDH1. But the new TCGA genomic analysis compares this glioblastoma and makes into the four subtypes: proneural, neural, classical, and mesenchymal. But what came out, the most fantastic thing from TCGA is they have identified three specific pathways which are activated in glioblastoma. One is the RB associated pathway, one is the P53, and the other one is the PI3 kinase pathway, RTK pathway, which is found in many glioblastoma. Actually, about 86% of glioblastoma have this activated pathway. And it also appears that due to the PTEN loss, this is a phosphatase loss, this pathways also can be activated also by the chromosomal 10q loss. And so the loss of this activates the glioblastoma's RTK-PI3 mTOR pathway, and how it works is the PTEN, which is a phosphatase sitting the right behind the PI3 kinase, opposite of PI3 kinase. And controlling the role of PI3 kinase in activating the AKT. But when PTEN is lost, AKT oncogene gets activated, and that activates plethora of cell signaling responses. And one of them, AKT activation, is to lead to the mTOR pathway. mTOR is multi-protein complex, very huge protein, and it makes, when it function, it makes two complexes, mTOQ1 and mTOQ2, and this is a serine-threonine kinase, which phosphorylate many other kinases in the downstream, and the important one is the S6K kinase, which we'll talk about, and the AKT is phosphorylated by mTOQ2. So when the S6K is down a lot, because it controls the negative, through the negative feedback, it controls the IRS pathway, but when it's too down, it breaks that control. So since it is so important in glioblastoma, there are multiple, the inhibitor of the mTOR, which is a rapamycin, and its analog has been tried in a preclinical trial for treatment of glioblastoma, rapamycin, and then the analog of this is a temsirolimus, and other have been made to be used as a clinical trial. Currently, this is a very short list. Currently, there are multiple these rapamycin like analogs are being used in clinical trials. None of the clinical trial have passed the phase 2 study. And one interesting clinical trial was done by the Clausi et al. And in that, they have used the P10 as a marker to choose the glioblastoma patient, because knowing that P10 negative patient would have the mTOR pathway activated, and then they treated the patient with sirolimus, and 2 times after the reoccurrence of first time and first surgery, and then after the second surgery. But to their surprise, what they found, that instead of the decrease in the AKT pathway, which it should decrease the phosphorylation of AKT, it has produced the activation of AKT via the loss of negative feedback, which was created by the S6 kinase downstream from the mTOQ1, and that enhances the another molecule which is controlled by the AKT is the PRAS40. So this trial was not only the molecularly very disturbing, but it also disturbing in terms of the clinical setting, as the patient who have this activation, they progress to the reoccurrence of the disease is the time was shorter than the patient who did not have this AKT activation. So that was disturbing. So what we did even before this study came out, that we treated the cell line of the glioblastoma with the mTOR inhibitor in a time response, and we saw the X6 kinase was decreased but not totally lost, but over the course of the time period there was an activation of phosphorylated AKT, and when we treated in this time dependent response, we see the migration of the glioblastoma cell, which is suppressed, but it over the course of time it actually increased. So that makes us think that it activates the another mitogenic pathway, which is a RAS-ERK kinase pathway, because over the time period of mTOR treatment, there is an activation of MAP kinase as seen here, and this MAP kinase is activated through the RAS-ERK kinase, because we also saw that the third bar, if you see the third, this is if we inhibit the mTOR by using the siRNA of mTOR. We see here in the middle panel, there is increase in the MAP kinase activity. And similarly, if the third panel shows, if we inhibit the mTOR, the ERK pathway using the ERK inhibitor U0126, there is no ERK activity. But when we give the rapamycin and the ERK inhibitor together, you start to see some activity reappearing. And since we wanted to prove that it's coming from the upstream pathway, so we use the MAP kinase inhibitor, and sure enough, we see as it activates the ERK kinase, phosphorylates the ERK kinase over the use of mTOR pathway, it also activates the MAP kinase, which is upstream of that. So what we found at that time, that this is the negative feedback, which was loop, which was controlled by the S6 kinase, and suppression of this to a level which activates the upstream IRS pathway, which activates the RAS MAP kinase, and the ERK pathway, and leads to the relentless growth and the migration. So that turns out to be that the rapamycin is an incomplete inhibitor of mTOR pathway, and it also activates the MAP kinase and mTOR1 pathway. So now what should we do? So we wanted to see how it affects, and if there any inhibitor which can suppress the PRAS40. Now PRAS40 is very interesting. The unphosphorylated PRAS40 binds with the receptor of the mTOR1 component, and it inhibits the activity of mTOR1 in an innate situation. However, if the AKT phosphorylates at the threonine 308 site, it activates phosphorylates the PRAS40 into 46 site, and that releases its binding with the receptor and activates the mTOR pathway. So this is the way the AKT and PRAS40 interacts here. So what we found was we used some other inhibitor than rapamycin, and we thought the small molecule inhibitor, which are ATP binding inhibitor. So we tend to use the taurine-1, taurine-2, and the XL388. XL388, a very small molecule, which is a promising result in many other cancers, and it's actually been used in treatment of renal cancer and some other. So this is the activity of the PRAS40, which is controlled by the AKT. And PRAS40 is a very interesting molecule. As you can see here, it has a multiple binding site, and it's right now is talked about the AKT mainly, but it appears that it also have some, the mTOR or directly also has some control over the PRAS40 structurally. So since the rapamycin and its analog have been unsuccessful in clinical trials, we decided to use the novel competitive ATP binding inhibitor, which can inhibit both complexes at the same time, mTOR1 and mTOR2. So we use this and examine the effect in inhibiting the glioblastoma cells, the stem cells. And you can see here how different they are. The rapamycin is an analog binding inhibitor for mTOR. It binds with the binding with the FKR12 protein, and that's how it inhibits the mTOR pathway. However, this ATP binding inhibitors, which are small molecule, they sit at the pocket of the mTOR at the ATP pocket and inhibits these two complexes. And in our study, our tumor population, when we did the activation of AKT and the phosphorylated mTOR, we found a high number of samples which is showing the activation of AKT as well as the mTOR at serine 2448 site. So we used this taurine 1, which was initially, and from there they made the taurine 2 and XL 388. And what we found was very interesting. The taurine 1 treatment over this is the dose response. You could see here taurine 1, taurine 2, both suppress the PRAS40 activity, but on the right-hand side, you can see the taurine 2 in the higher doses is completely abolished the activation of the PRAS40 and its effect on mTOR1 is reinstituted. So this is the effect. But when we do the other markers of mTOR pathway like 4EBP, that's down completely by the both mTOR1 and mTOR2. So PRAS40 to this inhibit the activity by the defined mechanism. Here is the PRAS40, which completely abolished by the mTOR2. You could see here, and but XL 388 did not inhibit the mTOR pathway. The downstream from the mTOR1 is the 4EBP1 was not affected by the XL 388. As you can see from here, the phosphorylation did not change, but the XL 388 did suppress the PRAS40 activity, but only at the higher concentration. You could see here. And when we did this, check the migration, cell migration, you could see that taurine 1 and taurine 2 both suppress the GBM cell migration, but the mTOR2 was more effective. XL 388 is absolutely did not change in the cell migration. They remained as the control cell migrated. So, and the cell proliferation was suppressed by the mTOR1 and mTOR2 and XL only in the higher doses. So the S phase entry, the cell cycle analysis also shows that mTOR1 and mTOR2 inhibited the S phase entry, but the XL 388 inhibited, but it was insignificant. And when we did the drug resistance analysis over the course of 5 weeks, you could see from here, the control cells never die. They keep on multiplying and producing more GBM cell. Rapamycin, you could see there is some apoptosis or cell death. Taurine 2 also caused the cell death, and XL was caused the cell death, but not over the course of 5 weeks. There is some less, however, it was significant. Now pay attention to the taurine 2. The taurine 2, all the tumor cells die. You can see here, the only surviving cells are the long elongated cells, and these are the stem cells. So basically taurine 2 was able to inhibit the tumor, the kill all the tumor cells, however, not the stem cell. Why stem cell is so important? So the cancer stem cells are the one which are involved in the origin of and maintenance of the cancers, and mTOR plays an important role, and especially in glioblastoma, this is one of the very important factors. As you can see, this is the as you can see here, there is a tumor. This is just the cartoon model. If you can kill the entire cell, entire tumor cell, but one stem cell is alive, that would regrow into the tumor. But if you can kill the stem cell, then the tumor would never come back. And in case of the glioblastoma, this is the model. I took it from Louis Paradas. You can see here the glioblastoma with the red and pink stem cell present in the tumor mass. And these are why they are different color. because they are genetically very heterogeneous. And sometime in a cancer, there would be the glioblastoma, there would be multiple different kind of genetically genetically aberrant cells are present, and they are very heterogeneous. So if we can kill this, then only we can be successful in treating. In our tumor population, the fresh tumor, we generated the stem cell, and this stem cell population shows all the property of stem cell, and they can be differentiated into the neuronal marker, such as the neuron and astrocytes. And so not only that, this is the real tumor area, which is not only tumor, but this is an extra tumor area, 3.6 centimeter away from the tumor margin. They are form the tumor. They are considered still a normal area. We find the astrocytes which are expressing reactive astrocytes, expressing the stem cell marker nestin. And it's very disturbing that the stem cell and the already altered astrocytes are present even at 3.6 centimeter away from the tumor margin. And when we do the immunofluorescent analysis, we see the nestin is expressed, mTOR is expressed, and in the right-hand side, the last picture shows the overlap of these. That means the mTOR somehow controlling the stem cell population of glioblastoma, because there is a strong overlap. And when we do the stem cell property analysis using the self-renewal test, what we find is that if you treat with rapamycin, the self-renewal goes down as compared to the treatment with the MAP kinase inhibitor. So we did the limited dilution assay, which is the assay to show the stem cell property. And here what we found was interesting. The red line is the control, and the you can see purple line is XL, which clearly inhibited the self-renewal. Green is taurine 1, but taurine 2 was not able to suppress the self-renewal of stem cell. That means it keeps the stem cell population, as we have shown by the drug resistant test. The stem cell is still there. And similarly, the growth of the stem cell are also altered by this compound. But look at the taurine 2, the blue, green, and red bar all fall into the 300 micron size. This is the size of the stem cell which do not progress further. They stay at that level only. However, you see control, the size is increasing, and even the 1000 micron size spheres are formed. Same as the XL388, the size is increasing, but the taurine 1 also dropped in size, but taurine 2 was more effective. So how do we do this? This is a very big question to target the stem cell. And what we did in my lab, we already published this paper, which is highly accepted. What we did, we treat the stem cells population with the differentiating agents. And after treating the differentiating agent, which can differentiate the glioblastoma cell into the three sub cell type, like neuron, astrocytes, and the oligodendrocytes, and then we treat the conventional way, and that's the way we can treat the stem, perhaps the glioblastoma. So in future, there may be these compounds such as the arsenic oxide or the other differentiating compound like all-trans retinoic acid can be used to treat the glioblastoma. So in the summary, the mTOR1, mTOR2, in the PRAS40 activation was suppressed by Turin 1, Turin 2, and somewhat by XL 388. Turin 2 suppressed migration proliferation more effectively than Turin 1, while XL 388 inhibited cell proliferation, but not the cell migration. The stem cell self-renewal and the neurosphere proliferation were suppressed by Turin 2 more than Turin 1 and XL 388. In conclusion, these results provide the molecular basis for the effectiveness of new class of combined inhibitors for mTOR1 and mTOR2 treatment. However, these are not these preclinical studies using this TORCI. These TORCIs are the one which are mTOR1 and 2 inhibitors, have provided a very poor responses. So in the disappointing efficacy in the xenograft model of glioblastoma. So this is and right now there are 3 trials. One is Abrupte. 2 trials is still going on using these combined inhibitors. So what do we do? So these were the second generation. Now there's a 3rd generation. These inhibitors have appeared. What they did, this is a very new stage. What they did was they take the rapamycin structurally and take the mTOR1 and mTOR2 inhibitor compound and link them by a linker and call them as a rapa link. This rapa link can, in a way, this can affect both the complexes of mTOR1 and mTOR2. And interestingly, this study was done in the glioblastoma xenograft model, and which provided an excellent results in suppressing the tumor, and also the authors claim that it can pass the blood-brain barrier, and so it's look very promising. And there are all the author. This paper was published in Cell, and all the investigators are from the very well institutions. But I was asked to write a report on that. This published in a Cancer Cell, and I made this picture. What they actually doing is this compound rapa link 1 is almost like a papayi, which can inhibit both the kinase side as well as the analog binding site of this mTOR pathway, and can inhibit these 2 complexes. And this paper was actually published in 2007, and let me know that there's no clinical trial going on for the glioblastoma. However, this rapa link has been used in clinical trial for other cancers, such as the prostate cancer. So in my opinion, for glioblastoma, the search for Achilles heel still goes on. This is the lab before the pandemic. This is the lab during the pandemic. And maybe Vito can recognize this Bernie Sanders sitting on the back. And this is the lab now this summer. So we're doing okay, and I'm done.
Thank you, Mina. Any questions?
Yeah.
화면을 띄워놨네. 화면을 띄워. 요거만 되지. 그래도 이렇게 된다니까.
There we go. I hope that's solved. So I confused, but it looked like in your activation assays, you were using hundreds of micromolar concentrations of Taurin 1 and Taurin 2. Oh, okay. Your slide's mislabeled. Because, yeah, I was confused as to,
It just came out as.
yeah, I didn't read it. I'm sorry.
Okay.
Yeah, okay. It was micromolar on the slides, and I thought that was very high, you know, so.
Yes.
I'm not sure I got this right, but I think rapamycin is working by competing with endogenous phosphatidic acid. Is this correct?
Is competing with?
Phosphatidic acid. The target of rapamycin is competing with phosphatidic acid for the mTOR complex to bind to membranes. And recently novel, and I think it is believed that the rapamycin problem is coming because phospholipase D enzymes are upregulated in cancers.
Yeah.
And recently, novel PLD inhibitors have been discovered. Do you have any idea if someone has tried to combine rapamycin with these novel inhibitors?
Not these inhibitors, but there are, rapamycin is currently, there are multiple trials is still going on with rapamycin to treat the glioblastoma in phase 1, phase 2, and they have combined with other inhibitors such as the VEGF inhibitor or the EGFR receptor inhibitor. So they combined trial has been going on, temozolomide and rapamycin. Rapamycin is still very popular in clinical trial for the glioblastoma.
Okay, thank you.
There is another, there is a question from the chat, and this is from Reinhold Schaeffer. Do these inhibitors trigger autophagy?
Wait, excuse me?
Do these inhibitors trigger autophagy?
I was asking this question because there's a link between MAP kinase inhibition and the stimulation of autophagy as a response. And I was just wondering if blocking, using rapamycin would have similar effects, because that would give a clue to also block autophagy and then kind of produce a better therapeutic effect.
Actually,
But it's speculative, of course.
that's very interesting question, but the way the rapamycin actually binds with the mTOR is very strong binding to the KB12, and that analog binding is considered to be very strong binding. I'm sure that with this another inhibitor, you say competing with that binding to the mTOR with the rapamycin.
I think he asked.
Oh, autophagy. Oh yes, autophagy.
Okay.
The downstream pathway of the mTOR1 is the one which controls the autophagy. And we have seen if you inhibit the rapamycin, there's increase in autophagy in the spinal cord injury patient. We just published a paper with the spinal cord injury, and the recovery is good with the rapamycin treatment because the autophagy increases. That a mechanism is controlling the secondary injury. But I have not seen in the glioblastoma. That's a very important question.
Yeah, there's this nice link in pancreatic carcinoma where MAP kinase inhibition plus hydroxychloroquine as an autophagy inhibitor produces a very good therapeutic effect, which even has motivated clinical trials.
Yeah, absolutely. That's a very important question. Yeah, we have not tackled in cancer, but we did in other disease.
Thank you.
Okay, thank you, Mina. We'll continue. The next speaker is George Carman. Please, George, the stage is yours.
Okay, hello, everyone.
Yeah, your,
I'm up.
yes, we see your presentation.
Great. So I'd like to thank Lucio for inviting me to share our story on the yeast PA phosphatase enzyme and its role in lipid metabolism. And second, I'd like to acknowledge the NIH for the support of our research. Our work centers on the regulated synthesis of the neutral storage lipid triacylglycerol, which consists of a glycerol backbone with fatty acyl groups attached at positions 1, 2, and 3. Triacylglycerol is stored in adipose tissue as a highly concentrated store of metabolic energy. We are all familiar with the obese phenotype when too much triacylglycerol is synthesized and stored. The opposite phenotype, namely lipodystrophy, results when too little triacylglycerol is synthesized. The synthesis of triacylglycerol begins with glycerol 3-phosphate, which may be derived from the glycolytic pathway. This molecule is subject to 2 successive acylation reactions at positions 1 and 2 to form the phospholipid phosphatidic acid. The phosphatidic acid is then subject to dephosphorylation to produce diacylglycerol, which is then acylated at position 3 to form the triacylglycerol molecule. The enzyme that catalyzes the penultimate step in this pathway is phosphatidic acid phosphatase. This enzyme, the subject of my presentation, will be referred to as PAP. The PAP reaction was discovered in 1957 by Eugene Kennedy and colleagues while working with chicken liver extracts. Our laboratory began its work on the enzyme from the yeast Saccharomyces cerevisiae in 1987. The image on the top right side shows yeast cells stained for lipid droplets, the organelle that stores the triacylglycerol. We have found yeast to be an excellent model system to study the PAP and its role in lipid metabolism. The role of the PAP reaction in lipid metabolism is depicted in this slide. When yeast cells are actively growing, the PAP substrate phosphatidic acid is primarily converted to the liponucleotide CDP diacylglycerol, which is then converted to all membrane phospholipids. However, when cells progress into the stationary phase, the PAP enzyme converts PA to diacylglycerol used to synthesize triacylglycerol. Now, mutants defective in the CDP diacylglycerol dependent synthesis of phosphatidylcholine and phosphatidylethanolamine can still synthesize these phospholipids from a diacylglycerol produced in the PAP reaction if they are supplemented with choline or ethanolamine. I also point out that the PAP enzyme plays an important role in controlling the cellular levels of phosphatidic acid, which mediates the transcriptional regulation of many phospholipid synthesis genes via the OP1, ENO2, ENO4 regulatory circuit. PAP activity is encoded by 4 genes in Saccharomyces cerevisiae. Each PAP enzyme has a distinct cellular location and function. The PAP enzyme that is involved in lipid synthesis is encoded by the PAH1 gene, and its reaction takes place at the nuclear ER membrane. The other PAP enzymes encoded by APP1, DPP1, and LPP1 are located at actin patches, the vacuole membrane, and the Golgi membrane, respectively, and they're involved in endocytosis and lipid signaling. I also point out in this slide that only PA1 is specific for phosphatidic acid, whereas the other PAP enzymes are promiscuous in their substrate specificity. As you might suspect, the PAH1 PAP enzyme is conserved throughout evolution. This slide depicts the conserved catalytic domains known as the NLIP and HAD-like domains in the yeast PA1 and mammalian lipin proteins. Also shown are phenotypes of lipodystrophy and neuropathy of mice lacking lipin-1 PAP and the obese phenotype of a mouse when lipin-1 is overexpressed. Additional lipid deficient phenotypes that are manifested in humans include rhabdomyolysis, insulin resistance, metabolic syndrome, and type 2 diabetes. Loss of PAH-PAP activity in yeast gives rise to a plethora of phenotypes that are summarized in the following slides. To begin, PAH mutants have elevated levels of the substrate PA and membrane phospholipids, and a lack of PAP activity in the mutant results in reduced triacylglycerol levels and the formation of lipid droplets. The elevated PA content gives rise to de-repressed levels of phospholipid synthesis genes, which contributes to the aberrant expansion of the nuclear ER membrane. In fact, the PAH1 mutation causes numerous other phenotypes depicted here. And so PAH1 mutants really suffer with an apoptosis phenotype and a loss of chronological lifespan. So as you might expect, PAH-PAP is regulated by genetic and biochemical mechanisms. Gene expression is induced as cells progress into stasis, and this is regulated by the transcription factors GIS1 and RPH1. Induction is also mediated by inositol supplementation via the OP1, ENO2, ENO4 regulatory circuit. In general, transcription is regulated in a manner opposite to that of phospholipid synthesis genes. Several mechanisms control the biochemical regulation of PAP activity. They include regulation by phospholipids, sphingosine bases, nucleotides, and by phosphorylation and dephosphorylation, and by proteasomal degradation. However, the most important regulation is via phosphorylation and dephosphorylation. Here I present a working model for the regulation of PAH. Expression of PAH1 is regulated during growth by nutrient status. After expression, PAH1 in the cytosol is phosphorylated by multiple protein kinases. Phosphorylated PAH1, indicated by the small gray circles, translocates to the nuclear ER membrane through its recruitment and dephosphorylation by the NEM1 SPO7 protein phosphatase complex. The phosphorylated PAH1 that is associated with the membrane catalyzes the conversion of PA to diacylglycerol, which is then acylated to form triacylglycerol that is stored in the lipid droplets. Dephosphorylated PAH1, or protein kinase C phosphorylated PAH1 that is not phosphorylated at the target sites for Pho85, Pho80, is degraded by the proteasome, as indicated by the dashed line arrows and ellipse. The domains, the regions, and the phosphorylation sites in PAH1 are shown here. The diagram shows the positions of the amphipathic helix required for membrane association, the conserved N-lip and HAD-like catalytic domains, a conserved tryptophan residue required for PAH function, the acidic tail that interacts with the NEM1 SPO7 complex, and the intrinsically disordered regions where nearly all of the phosphorylation sites reside. The serine and threonine residues known to be phosphorylated are grouped at their approximate positions and marked for the responsible protein kinases that have been identified to date. The phosphorylation of PAH is complex, which is mediated by multiple protein kinases. Some phosphorylations are hierarchical in nature, meaning that the phosphorylation at one site affects the phosphorylation at another site. And some are redundant, meaning that multiple protein kinases may phosphorylate the same site. Work is ongoing to identify all the protein kinases involved. To simplify, the phosphorylation attenuates PAH function, whereas the dephosphorylation stimulates function. The remainder of my talk will focus on the conserved tryptophan residue that is required for PAH1 function. Through a series of mutagenic studies, Young-Hee Park discovered a WW domain, shown here in red, that lies within the C-terminal intrinsically disordered region. This domain is required to complement the temperature-sensitive phenotype of a PA1 mutant. To put things in perspective, you can see that the catalytic N-lip and HAD-like domains are also required for PA1 function, as their deletions result in a failure to complement the temperature-sensitive phenotype of a PA1 mutant. In this slide, I show the WW domain is conserved in yeast and mammalian PA1 and lipid proteins. Young-He constructed mutants for residues within the domain, and only those for TRP637 failed to complement the temperature-sensitive phenotype. So we focused on that amino acid. Young-He examined the effect of a W637 alanine mutation in PA1 on lipid composition and lipid droplet formation. In this experiment, the wild type and the mutant were expressed in cells that possess the NEM1 SPO7 complex. The cells were labeled to steady state with C14 acetate to label the lipids. And when the cells progressed into stationary phase, the phase of growth where PA1 has its greatest effect on lipid metabolism, the lipids were extracted and analyzed by thin layer chromatography, followed by phosphorimaging and image-borne analysis. And the levels of the individual lipids were normalized to the total radioactivity in the chloroform soluble fraction. In these cells expressing the mutant, the amounts of diacylglycerol and triacylglycerol decreased by 2-fold and 6-fold respectively when compared with the wild-type control. At the same time, the amount of phospholipids in cells expressing the mutant were 1.4-fold greater than that found in cells expressing the wild-type enzyme. These results are consistent with previous work showing that PA1 functions to control the bifurcation of PA between the synthesis of triacylglycerol and membrane phospholipids. The images on the right side show the decrease in diacylglycerol and triacylglycerol correlates with a decrease in lipid droplets, the organelle that contains that lipid. We considered that the reduction in triacylglycerol content and altered lipid composition of cells expressing the mutant enzyme is rooted in a defect in PA's ability to associate with membranes. To address this question, the wild type and tryptophan mutant forms of PAH1 were expressed in cells expressing the NEM1 SPO7 complex, followed by an immunoblot analysis of the protein in the cytosolic and membrane associated fractions of the cell. As expected, most of the wild type protein was associated with the cytosolic fraction, with a barely detectable amount associated with the membrane fraction. This analysis showed that the tryptophan mutant did not impair the membrane association of PA1. In fact, a greater amount of the mutant protein was associated with the membrane fraction. We considered that although the tryptophan mutant protein associates with membranes, it may associate with the wrong membrane. The translocation of PA1 to the cytosol to the membrane fraction requires its recruitment to and dephosphorylation by the nuclear ER membrane-associated NEM1 SPO7 protein phosphatase complex. And to examine whether the association of the tryptophan mutant fraction is mediated by the phosphatase complex, the mutant was expressed in cells lacking the complex. This analysis showed that in the absence of the complex resulted in a discernible reduction in the amount of the tryptophan mutant protein associated with the membrane fraction. Thus, translocation to the wrong membrane is not the basis for the mutant phenotype. We examine the effect of the tryptophan mutation on the translocation of PA to membranes by an in vitro approach. In the experiment shown on this slide, Jordan Stuckey mixed the cytosol fraction that contained hyperphosphorylated PA1 with the membrane fraction from cells expressing the NEM1 SPO7 complex. Following a 30-minute incubation, the mixture was centrifuged at 100,000 times G to re-isolate the membrane fraction. As you can see, the wild-type protein translocated to the membranes. In agreement with the in vivo localization data, the tryptophan mutant did not show a defect in translocation. In fact, a greater amount of the mutant protein associated with the membranes when compared to the wild-type control. Moreover, when the PA1 translocation assay was performed with membranes derived from cells that lack the NEM1 SPO7 complex, neither the wild-type nor the tryptophan mutant protein translocated to the membranes. In sum, the tryptophan mutation does not impair the NEM1 SPO7 dependent translocation of PA to the membrane. Because phosphorylation is so important for the regulation of PA1, we question whether the mutation affects the endogenous phosphorylation of the protein. For this experiment, the hyperphosphorylated wild-type and mutant proteins were isolated from cells lacking the NEM1 SPO7 complex. The proteins were subjected to a mass spec analysis of phosphorylation sites. Collectively, the mutation caused increases in the phosphorylation of sites within the intrinsically disordered regions at the N-terminal region and decreases in the phosphorylation of sites within the intrinsically disordered regions at the C-terminal region. The decrease in phosphorylation at the C-terminal region is consistent with the greater translocation of the mutant enzyme to the membrane. However, the effects of phosphorylation and dephosphorylation are complex and difficult to explain in a definitive manner. We question whether the PA activity of the membrane-associated tryptophan mutant of PA is defective. To address this question, the wild-type and tryptophan mutant forms of PAH1 were expressed in cells with an APP1, DPP1, LPP1 genetic background. This genetic background was necessary to eliminate the competing PAP activities encoded by the APP1, DPP1, and LPP1 genes. This analysis showed that the PAP activity of the tryptophan mutant was not defective. Actually, the membrane-associated PAP activity in cells expressing the mutant was fourfold greater than the activity in the membrane fraction of cells expressing the wild-type protein. The greater activity in the membrane correlated with greater association of PA with the membrane that it was in its dephosphorylated state. On the right side of the slide is an experiment with purified, unphosphorylated enzymes that shows that the tryptophan mutation doesn't affect the inherent PAP activity of the enzyme. Insight for the role of TRP637 in PA function might be obtained from a structure of the enzyme. However, the unfolded nature of PA1 has precluded its structural analysis. A similar situation has precluded the structural determination of mammalian lipid proteins. In the absence of experimental data, a computer-generated structure based on the AlphaFold program shows that the NLIP and HAD-like domains, the catalytic ASP398 and ASP400 residues within the HAD-like domain, the N-terminal amphipathic helix, and TRP637. As you can see, the TRP637 aligns with the catalytic residues and the amphipathic helix, and we hypothesize that TRP637 plays an important role in positioning the enzyme with PA head group at the membrane surface. The alignment of the conserved tryptophan with the catalytic residues is conserved in the model of human lipin 1. To summarize, the PA-PAP enzyme plays an important role in lipid metabolism. The enzyme is regulated by genetic and biochemical mechanisms, with phosphorylation and dephosphorylation being most important. A conserved tryptophan was identified that plays an important role in PA function, but the tryptophan mutation causes a loss of function has yet to be discovered. It doesn't impair the cytosolic to membrane translocation or the PAP activity in vitro. Computer modeling suggests that TRP637 plays a major role in positioning the enzyme with the active site at its substrate on the membrane. Finally, I would like to acknowledge the Carmen Laboratory family members who have contributed to this work, and also thank you for listening to my story. Thank you very much.
Thank you very much. Any questions?
Hi, George Vitas.
Yes, thank you, Vitas.
So,
I had to jump on to shut on the lights.
right. So my question is actually pretty simple. The W to A mutation is pretty drastic, and tryptophan is often involved in sort of stacking interactions, you know, either within a protein or with binding partners. And this tryptophan sits in a intrinsically disordered region, right? Is that correct?
Yes.
So have you actually looked to see if, since this is predominantly cytosolic, if you just take yeast lysates and you filter them, do a gel filtration, and essentially ask, you know, do you actually have a larger particle or a different particle in the mutant than the wild type? Because in intrinsically disordered regions, as you indicated, these are oftentimes modified and then they become coat hangers essentially for binding partners. And I don't think you addressed that possibility in the talk.
Yeah, well, if you look on the Saccharomyces genome database, 47 different binding partners have been identified with PAL1. And it turns out that all of those binding partners are the protein kinases that phosphorylate the enzyme in the cytosol. So the only binding partner that has been identified with the membrane is the NEM1 SPO7 complex. So yes, we had considered that.
Hi, Nick speaking. Hi, George.
Hi,
Nice to
Nick.
meet you. You have shown that the different PAP have different subcellular localization, but does anyone know if they have preferential substrate depending on the fatty acid composition of the PA?
Yeah, we've done studies like that, and there's no specificity for the fatty acyl moieties of phosphatidic acid, right? And we've also looked at that in with the human lipin protein.
Okay, thank you.
So it seems that the enzyme only, you know, recognizes the phosphate head group.
I have a question via chat. I can repeat it.
Okay, is membrane associated? No, not that way. Is the membrane associating surface of PAP? No, not that. It's the amphipathic helix is involved in the association immediately following dephosphorylation by the complex. But then the rest of the protein, you know, must associate with the membrane, and those other sites have not been mapped out.
So do you think the TRIP residue has a direct role or it facilitates somehow the positioning of catalytic residues that also need to be in the proximity to the membrane?
That's the hypothesis. You know, we have not proven that.
How do you think this hypothesis can be validated experimentally?
Well, it's really difficult without having, you know, the actual structure for it. You know, we've considered making peptides and looking at peptide, you know, membrane association, but I mean, that's one thing we've thought about, but we're really stuck. We've been working on this for a number of years, and everything that we expected would be the answer never turned out to be the answer. This is the George Costanza of lipid metabolic enzymes. So
okay. So NMR, solution NMR spectroscopy might be quite useful for characterizing the conformational preferences of IDRs and their propensity to associate with membranes.
Yeah.
Thank you.
Thank you for your suggestion.
Okay. Any additional question? Okay, if this is not the case, thank you again, George, for this. And we'll continue to the next talk and the last talk of the day by Daniel Raven.
Okay, I'm going to share my screen. So I realize I'm between you all and dinner, so I'll try to make this an efficient talk. I'm going to go through some slides in the beginning that just sort of reviews what we've done before, and then I will spend a little more time on what some of the new findings that we've got about this enzyme. So we've been looking for a number of years now at this enzyme called diacylglycerol kinase theta. And the enzyme catalyzes the phosphorylation of diacylglycerol to phosphatidic acid, which George was just talking about. The back reaction was the enzyme that George was talking about and done a lot of work on, was the PA phosphatases. But we've been focusing a lot of our attention on the diacylglycerol kinases themselves, the ones that convert diacylglycerol into phosphatidic acid. And this is kind of important because, as this slide here shows, that the diacylglycerol and the phosphatidic acid both have very strong signaling roles. And some of them have been shown, or at least strongly suggested, to be regulated by this enzyme, diacylglycerol kinase. For example, PKC is regulated by diacylglycerol, and the conversion of diacylglycerol to phosphatidic acid turns off a PKC. So there have been a number of proteins that have been regulated either by diacylglycerol or phosphatidic acid, and the generation of those molecules, or at least the level of those molecules, are modulated by diacylglycerol kinase, or at least one of the isoforms. There are actually 10 mammalian isoforms of diacylglycerol kinases. They're in 5 different classes. They have various biochemical signatures to them. And the one that we've been looking at for a number of years now is the type 5 diacylglycerol called diacylglycerol kinase theta. It's stimulated by phosphatidylserine and phosphatidic acid. And it's also stimulated by proteins with a polybasic region, which I'm going to talk more about. And it's inhibited by GTP-bound Rho A. So we noticed a number of years ago, we showed that proteins that have a polybasic region will activate this enzyme. And that's shown here, where if you isolate the enzyme in vitro, I get a purified enzyme, which took a little while to do, and then ask whether it was activated by a series of proteins. And the reason why we did this is because when we found the enzyme in an isolated form, it had very little specific activity. But when we added various proteins with different amounts of basicity to them, we got an increase in activity. And you can see here, polylysine and polyarginine both activate the enzyme, whereas free arginine and free lysine do not. Cytochrome C has a considerable amount of basicity to it, but those basic residues are not accessible, so it doesn't really activate the enzyme. And we did some enzymology on this. Two of the proteins that will activate the protein, or one of them in particular is tau, and it has a K activation of about 24 nanomolar. We looked at tau because originally we thought tau was an endogenous activator, but I don't think that's true anymore. But what is important is that these activators actually are important for the activation by the lipids I just mentioned. So for example, phosphatidylserine and phosphatidic acid activate the enzyme, but that activation is seen in the presence of a polybasic protein activator. That's true for phosphatidic acid as well. In this case, we use polylysine. So one of the first questions that comes up is, do the activators interact directly with the enzyme? And in fact, it does. You can show this by co-immunoprecipitations. If you immunoprecipitate diacylglycerol kinase theta and look for the presence of an activator such as tau, you can see the immunoprecipitation. If you immunoprecipitate tau, you can see the interaction with TGK theta. This is done in vitro with purified proteins. So we were very certain, at least at this point, that the enzyme interacted with the activator directly. But even more so, recently what we've done is we've shown that if we use a soluble substrate, a diC6, we can show that the enzyme can be activated by a polybasic protein. In this case, it's polylysine. So this is just showing the concentration dependence of the diC6, and this is showing the difference in activity when in the absence and presence of polylysine. And initially, we were kind of concerned by this, although we just heard a talk where someone pointed out that a polybasic region is important for an in vivo interaction with a protein. And it turns out that in vivo, these basic residues are important, as particularly in a cellular context. For example, the casein kinase 2 can be activated by a variety of polybasic compounds. Can you guys see this slide okay? I'm assuming yes. So it shows the same kind of profile that we see with our enzyme being activated by a variety of polybasic proteins. But it turns out that in vivo, this CK2 is activated by the basic N-terminus of an SK2 channel only when the channel is closed. So the cellular context of the activation is very important. So this is a little comforting to us, because at least we started to realize that the cellular context may be important. And we looked at some enzymology of this, and what we found was that the activator actually had a twofold effect on the Km for ATP of the enzyme. It actually also appears to lower the Km of the surface Km for the substrate, diacylglycerol. It was a little difficult to tell because we couldn't get the enzyme to saturate without the presence of an activator. But it appears to have an effect both on the Km of the diacylglycerol, this is in a liposome here, as well as on the Km for the ATP. So overall, diacylglycerol kinase is activated by polybasic regions of proteins. The activation doesn't require membrane binding, and the polybasic regions appear to lower the Km for ATP and the surface Km for the diacylglycerol. So one of the things we got very curious about is what roles this enzyme played in the central nervous system. And the reason why is because in C. elegans, when they knocked out the homolog of this protein in C. elegans, there was a constitutive release of acetylcholine at neuromuscular junctions. So we looked at this and we kept in mind what happens during the synaptic vesicle cycle. So during the synaptic vesicle cycle, synaptic vesicles are actually up here as well, but they dock on a membrane, and then they sort of secure themselves to the membrane, if you will. Then this is called the readily releasable pool. And then when an action potential comes along and calcium flows into the neuron, in this case, there is an exocytosis where the neurotransmitters are released. And that exocytosis has to be followed by endocytosis that's called the compensatory endocytosis, because if this didn't happen, the bouton at the neuron would get so big it wouldn't be very functional, or it would be dysfunctional. And here what we're showing is that there's a variety of lipids that have been shown to be involved in this path, in this cycle: diacylglycerol, phosphatidic acid, as well as some of the phosphoinositides. And these things are thought to be important because of their interactions with various proteins. For example, dynamin 1, which is very important in endocytosis, interacts with phosphatidic acid, and there are others as well. So we decided to see what role DGK had in this process of diacylglycerol kinase. And what we did was we first used an assay, which is called a synaptofluorine assay, where synaptofluorine is linked with a fluor that is sequestered or quenched in the acidic vesicle. And when there's an action potential that fires and those vesicles fuse with the membrane, there's an increase in the fluorescence that can be observed on the neuron. And then as the vesicle is taken back up, which has a half time of about 34 seconds, it is then rapidly acidified, and that you lose the fluorescence. So you can see that there's a rapid increase and then a decrease during this exo and then endocytosis. Well, we looked at these knockout mice that had the DGK theta knocked out, and what we found was there was a slowing of the compensatory endocytosis. That's shown here. And this is quantitated here, where you can see in the wild type enzyme, that is wild type neuron, the wild type neuron, and the wild type neuron expressing a wild type DGK theta, there has no real effect on the endocytosis rate. And when it's also transfected with the kinase dead diglyceride, it doesn't really have an effect on the endocytic rate. But if you look at the knockout mice, it has a really lengthened endocytosis rate. So it's kind of slows endocytosis. That's recovered when we co-express, or when we express the wild type enzyme, but not when we express a kinase dead enzyme. This is showing the localization of the enzymes here. So this says that the diacylglycerol kinase is important, and the catalytic activity of the diacylglycerol kinase is important. And what's particularly important and interesting is that this defect, it actually worsens as you increase the number of stimuli, or if you stimulate the cells with higher frequencies. So this is particularly important. This is shown here. This is particularly important that if you're in some kind of epileptic, say, a kind of situation, that would actually cause a rapid increase or a larger increase in the difference between wild type and knockout enzymes. It's also important in something else I'll talk about if anybody's interested. It happens in neurons called that fire all the time, that fire continuously. So in regular neurons, you get a release of neurotransmitter when you stimulate the neuron, and that usually results in a spike of the activity. But in cases like in your eye or your ear, where you have to respond to light or hearing in a sort of linear fashion or more of a linear fashion, there's a continual release and a continual increase and diminishing of the exocytosis. This defect is seen at a greater level, even in the wild type cells. And I can show you that data if anyone's interested, but we've done that with retinograms in the eye. So this was all very interesting. But the next question was, what's the protein in the central nervous system that's regulating DGK theta? And this is where it got kind of interesting, because we really didn't know what protein with a polybasic region could possibly be doing this. So one of the things a graduate student in the lab did was she did proximity labeling using APEX2. And what she did was she hooked up the apex to a GFP diacylglycerol kinase, and she expressed it in neurons. And then what she showed, first of all, was that in order to get any labeling with the apex, she needed a complete mixture, whether the neurons were depolarized or not. If she eliminated anything, the biotin phenol or any of the reagents, there wasn't any labeling. She also showed that the DGK theta was actually expressed throughout the neuron, but it was also found in the same place as V-GLUT1, which is the presynaptic region of the neurons. So that's what really set things up. So we were able to actually convince ourselves that we had a system where we could look at the proximity labeling of these of the interactors of DGK theta. So and of course we also showed that the DGK theta itself was labeled, which was important because it had the APEX2 on it. We felt that it should have been a good internal control, and it is. So when we looked at this, we found that there are a number of proteins that are labeled or increase in labeling when the cells are depolarized, and some that actually decrease when the cells are depolarized. And this is just a table of 3 experiments that we did showing the total number of biotinylated peptides and the total number of biotinylated proteins, because they're trypsin digested before they're analyzed. And this is just a sort of a pie chart or an indication of the number of proteins that overlap in all 3 experiments. And you can see that approximately 50% of the proteins were reproducibly biotinylated in the 3 experiments. Some were not as reproducibly biotinylated, but at least 50% of them were. So the next thing that we did in analyzing these things was we wanted to validate the biotinylations. And one way we did that is by a streptavidin pull down. So we biotinylated the proteins and the peptides, and then we passed them over a streptavidin column and eluted them, and then did a Western blot. And one of the things that we noticed was that there's at least these proteins that we're indicating here were all validated by the streptavidin pull down, but one that caught our eye in particular is synaptotagmin 1, and I'll explain that in a second. Synaptotagmin 1 is biotinylated under these conditions and is validated by the streptavidin pull down. It's also validated when we did co-IPs. We did this to further validate it. So what we showed was that if we just co-IP'd the DGK theta or the interactors, synaptotagmin 1 also co-IPs with the diacylglycerol kinase theta. Now this was interesting to us because I'll point out now that it had a pretty significant basicity to it, about 15% basicity to it. It is a synaptic vesicle Butan protein, and its function has been shown to be coupling membrane fusion exocytosis primarily. That's what it was originally shown to be involved in. And I should point out that while I mentioned before that DGK theta is ubiquitously expressed throughout the neuron, we focused on proteins that are in the synaptic Butan. We focused on proteins that are particularly in the presynaptic region, just to kind of focus where we were focus our efforts because we knew it was somehow involved in compensatory endocytosis. So there's more than these proteins, but these are the ones that we focused on. So these are the ones that are in either the bouton or the vesicle that are likely involved in synaptic vesicle cycling. So we took all of these proteins and we asked the question that many of you are probably asking yourselves is, do these proteins activate the enzyme in vitro? And the answer to that is yes. And in fact, synaptotagmin 1 does an excellent job. It's often about 15 to 20-fold activation over the enzyme alone in an in vitro assay. The other proteins often do some activation of the enzyme, and a couple of them, Dynamin 1 and Monk18 in particular, don't do very much at all. But synaptotagmin 1 really did a whopping good job of activating this enzyme. And this is shown just in a little more of the specific activity changes. This is with no activator present. This is with DGK theta and 50 nanograms of synaptotagmin 1, and this is 500 nanograms of synaptotagmin 1. And you can see that it is dose dependent or concentration dependent, at least in this case. So why did this catch our eye? Well, this caught our eye because there was a report from Ed Chapman's lab a few years ago that showed that synaptotagmin 1 was involved in both exocytosis and endocytosis. So this is where he's showing that two regions of the synaptotagmin 1, which bind calcium, they're called C2A and C2B. These regions bind calcium residues, and they actually the mutation of these residues, mutation of the calcium binding portions of this actually can rescue, if you will. I mean, not rescue. They actually can cause the same phenotype, but if you only mutate one of those calcium binding regions, or in the C2A or the C2B, you can rescue the phenotype in the synaptotagmin knockout neurons. So these neurons are knocked out with synaptotagmin, and then you can see that you can rescue it by coupling in, putting back in either C2A or C2B. So either one will rescue it. And this figure here looks a lot like the figure we got from the endocytosis. So this actually has generated a model that we currently have where we think that these vesicles which have synaptotagmin on them, when a calcium spike comes in because the neurons are firing, there's a action potential. Part of the synaptotagmin is involved in binding to the membrane and assisting in exocytosis, but part of the synaptotagmin is also interacting with DGK theta and to activate DGK theta, generating a PA that's important for endocytosis. And in a little more detail, what we think might be happening is that at this region where the endocytosis is occurring, that's where we believe, and we haven't quite proven this yet, where the DGK theta is actually being activated by the synaptotagmin. This would actually provide for a phosphatidic acid pool that could bind to the dynamin, which is important for the pinching of the endocytic vesicle. So overall, we think we're getting very close to understanding that the endogenous activator, at least the endogenous neuronal activator involved in SV cycling, is synaptotagmin, and it's found on the vesicle, that synaptic vesicle that's coming into the membrane of the bouton for exocytosis. Now, the final question is, what's the molecular mechanism of all this? And this is a work that's still in progress, and what we're very much in progress. And what we've been doing is trying to get a structure of this enzyme, which has been very difficult. You can see here, just in terms of what it is known about some of the structures of DGK theta, of DGK enzymes, there isn't any full-length 3D structure of any of the eukaryotic DGKs, but there is some information known about some of the domains. For example, the EF hands, the SAM domain of delta. There is a DGK B. This is a prokaryotic enzyme, which is actually just a catalytic domain, because that's all it is. And in DGK delta, the C1 domain actually has been, the structure has been worked out. So we've been taking the rather risky task in a way of trying to elucidate the structure by cryo-EM. And I got a very persistent and and very hardworking postdoc on this. And she's been able to get some resolution using via cryo-EM that we've gotten down to a structure that's about 8 to 9 angstroms. And we've got to get at least below 6 to see any regions that would really relate to an alpha helix, for example. But we're starting to get an idea. She modeled in sphingosine kinase 1 because it actually fits better than the prokaryotic DGKB. And what she showed, and I'm going to show that this is the catalytic domain, and we believe this is the accessory domains. This is the RAS binding domain. This is the C2, one of the C1 domains, and this is another one. I think the C1 domains. This is probably a very flexible region that we just can't map out. You can see that the dynamic nature of this protein is actually what gives is inhibiting, I should say, or reducing our ability to get a really good smooth curve in this, what's called a Fourier shell correlation, to really nail down what the resolution is. But we're working on trying to make this a little less dynamic so we can nail that down. But when you look at the structure, and this is an alpha fold structure, I'm sorry, this isn't the alpha fold structure. This is the magenta here is the known structure of sphingosine kinase 1. So we have the sphingosine kinase 1 structure. And when we model that in to what we know or what we think is DGK theta from the alpha fold, we get a very close alignment of SK1 with DGK beta. And that allows us to get an idea of where the lipid might be binding the diacylglycerol, as well as where the ATP is binding. The ATP actually is being held in there by part of the C lobe and part of the N lobe. So there's an N lobe and C lobe to these catalytic structures. So we're making some progress on this. We haven't quite gotten down to the structure we want, but we think we now have an idea of what is regulating this enzyme in the central nervous system. And we think we're getting very close to the structure. And so we're going to be able to eventually model where that synaptotagmin, if it is synaptotagmin, where it binds to the diacylglycerol kinase theta, what it does to the structure to generate the phosphatidic acid. So with that, I'm just going to thank the people from my lab and Sandra Gabelli's lab and Mario Ams's lab and Rick Hugener's lab. Rick is a neuroscientist. Becky Tusakina, Hannah Goldschmidt, Elizabeth Petro. Chen Ma is the person doing all the cryo-EM, and she's being assisted by Duncan Sosa. Casey Barber is a graduate student who's getting ready to graduate, who's has been doing the Apex labeling. And so I'll stop there and take any questions you might have.
Thank you, Dan, for this nice summary of your work on the DGKs. Any questions? Here I see here in the lecture.
I think there might be one in chat. No?
Hi, Dan, Nick. Very vivid presentation, nice work. Do you know if, did you check actually the morphology of the spines that the boutons that you're looking? Because we have used the DGK kappa knockout mice and the spine look very differently.
Yeah, that's an excellent point, Nick, that we don't see a very big difference in DGK theta on the spines. I think that there are other isoforms, as you're pointing out, that actually do seem to play a role in spine density or spine morphology, but we actually took a look at that. We didn't see any effect of knocking DGK theta out on the spines. We're happy to send you the construct if you like it to test it, or the other DGK knockouts that we have, but
sure.
But we didn't see any effect.
Okay, thank you.
So I have a question. Your cryo-EM, you docked in one of the lobes, I guess, where the lipid binds, but the two lobes of the protein are not in the same size. However, the structure that we're missing is more or less in the same size that the structure that you docked in. So how can you explain that?
Yes, but actually the N lobe and the C lobe, in that there's a cleft where the ATP is kind of more in this, being held by the C lobe and the N lobe, and the DGK is coming in from the N lobe. So this is all conjecture because we're just docking it. And and we think that while they're not exactly as close as we think they are, at least in the docking, we think there's a phosphotyrosine actually intermediate. It sort of reminds us of the phosphohistidine in PLD activity that may be mediating the transfer of that gamma phosphate. So you make a very good point. We just don't know, but we think that there may be a 2 or 3 step, at least a 2-step mechanism.
Can it be a dimer?
See a what?
Can it be a dimer?
Oh, now that's another excellent question. So we've been playing with this a little bit because when we purify the enzyme and we run it over a sizing column, we do get some fractions that look like a dimer. We're trying right now, we're trying some glutaraldehyde so we can get more of the dimer so that we can see more of it on the cryo. Because, the cryo you get like, a million billion particles, 100,000 particles, you get down to maybe 50,000 or 10,000 you can actually use, and then you try to class that. So we've got to, it's a small percentage of the total particles that you can actually use. So we're trying to enhance the number of dimers and maybe see that, or generate dimers artificially, because if we can get something that's a little larger than 120 kilodaltons, we'll have a much better chance of seeing something in the getting better resolution in the cryo. So that's a work in progress.
Thank you.
So Dan Vitas, first of all, congratulations. This story has come quite a long way in the last couple of years. So I've got 2 very quick questions. The first one is, the presynaptic terminal is a crowded place. So have you used, and since you were doing this in cultured neurons, have you used SILAC or some other mechanism to actually just basically assess what your ratios are of biotinylated protein in your test case, versus a control? And that might actually tell you true proximity, because, you have these zones of splatter, biotin splatter, and you're going to pull them all down, which probably has happened.
Yeah. No, that's another excellent question, Vito. We've talked about SILAC. God, I'm blanking on his name. Pandy, Akhilesh Pandy was in our department for a while, and we started working on that with him just before he left. But we have been thinking about doing SILAC. The other thing we're thinking about doing with Jay Shao is a super resolution microscopy to see if we can show that any of these proteins, particularly synaptotectin and DGK, show a greater co-localization with each other, interaction with each other, actually during an action potential. So yeah, that's a good question. That's a good thing. We just haven't done it yet.
Yeah. And the last question is obviously you have the knockout mouse, right? You have the knockout mouse or knockout neurons. And so the issue is you didn't mention what the phenotypes are. And if they're mild, do you have how shall we say, a friend of DGK theta that might be compensating?
Yeah, that's a very good question. Another very good question. So in the neurons, there are, I think, at least 9 of the DGKs that are there, and they don't compensate the endocytosis defect. So there aren't moonlighting. However, you ask a very good question. So we have the DGK theta knockout mice that I'm willing to send to Nick here if you'd like them. But the what we did notice was that when you and I didn't show this data, but when you do the electroretinograms, these mice appear to have a defect in what's called the bipolar cells in the retina, and they're slowed down, and so we think these mice are night blind. And we have some other behavioral evidence that actually might be true. And the reason why that's interesting is what I said before, was that it's in the retina where there's a constant stimulation or exocytosis and endocytosis in the neurons that's gauged or varies with the amount of light that's coming in. So they're constantly being stimulated at more than just a basal level. So there is a phenotype, but we think that at least one of the phenotypes is going to be in sensory neurons like that. Whether there's other phenotypes, we're not really sure yet. We haven't really done behavioral studies, so we don't really know, but we do think they're more aggressive.
Okay, thank you. Olfaction, that might be one to look
Yeah.
at.
Right, and hearing.
Okay, I don't see additional questions, so I would like, let's thank Dan again. And all the speakers in this fantastic session. And I would like to remind you that we are reconvene here at 2 o'clock Bologna time. The rest of the world is less of importance, but and I wish you a great morning, evening, night, and here a continuation of wine session and dinner.
I wish I was with you all.
All right.